Please,
Okay,
the stage is
thank you.
yours.
Well, it's really a pleasure to be here. This is a special occasion. I was really taken by all of the beautiful speeches last night. We had to leave a little early because I needed to get some sleep. I'm going to start by just a handful of reminiscences about some interactions with Michele that I think he'll remember. In 2012, and it's hard to believe 13 years ago, he very generously invited me and my wife at the time to come there for an extended stay in Lugano. And we did some things. This is a paper that Michele wrote with a very brilliant graduate student I had that came with me, if you recall this. And this, you can argue whether this is a proof or adds to the proof or adds information, but this really was a very clever piece of work, and I know that Michele had a real part in working with James a lot on this. It was mapping well-tempered metadynamics into a very interesting set of differential equations. And the important thing is you could show that it really would converge asymptotically to the exact result. And that led to a spin-off, which, having understood sort of a new perspective on the metadynamics, if you had a knowledge of the beginning and end point, so for example, a reactant and a product, right, you knew what they were, you could recast metadynamics into something called transition-tempered metadynamics. Something like ATP hydrolysis in a protein, which is, it's a holy grail, right? ATP, GTP hydrolysis reaction is very hard, and you would use QM-MM, but if you want to do it right, you would never just look at a minimum energy path. You'd want to do free energy sampling, and likely in more than one collective variable. And transition-tempered metadynamics that basically came from this work with Michele allowed us to do that. These are free energy pathways along two essential CVs for ATP hydrolysis. This is for the monomer, this is for the filament. You can trace out the minimum free energy pathways, and if you look at these and compare it to experiment, the delta G, it's really right bang on. And this was a few years ago now, but it was a huge demonstration of what could come when you mixed, QM, you call it ab initio MD or QMMM with enhanced sampling, and you just need the right degree of efficiency. And I'll just say, Garnett Chan and I are working now on speeding up the quantum part, so that the ability to study this incredibly important process in biology will be, I hate to call anything routine, but it will be done, and it's really, really very, very important. So this came from spending a couple months with Michele. I think we wouldn't have done it otherwise. Okay, coarse graining. I've shown these view graphs many times. Years ago, so 20-some years ago, coarse graining was beginning to be done. I don't know if Mike Klein's here. He was doing some interesting work. Marink was just published a paper, and it occurred to us, what exactly is this, and can we formulate it from a point of view of statistical mechanics? And of course, statistical mechanics is you start from molecular information and you push up in scale to something. And coarse grain modeling is, representing molecules as a simpler objects, and some bottom end point. How you do it rigorously is you need mapping functions, mathematical functions that map the atoms into these beads. We call them beads for reasons I don't know. Some simple manipulation shows that if you could do this, the interaction will be a potential of mean force, right? A free energy surface of many, many dimensions still, because there's a lot of coarse grained particles, and you just can't do this. It's impossible. We do it now for a few degrees of freedom. So again, the touch of Parinello. It so happens, which I mentioned this in words when I give this talk, but I want to show you explicitly. There was a really wonderful guy, Sergei Izvekov, that had come to me as a postdoc and then went to work with Michele. And we published this paper on taking ab initio MD and doing force matching. We didn't invent force matching. I think Ercolesi and Adams did this. But this whole methodology of doing it numerically, sort of machine learning, basically, right? You could map ab initio MD simulations into these force fields, and Sergei, to his credit, really figured out the numerics of that. And that existed. So then Sergei came back to me, and by the way, Michele will remember, editing one of his papers, the English was unbelievable, right? I mean, I think he even told me he just couldn't do it. I mean, it was eventually, but he was really smart, so it was worth it.
I
every other word, right?
But that was the absolute minimum.
It's much better now, much better. But anyway, Sergey was really smart, so he did this. So this is where someone mentioned last night, insight is important before the mathematics and everything. It occurred to me, so the core of the force matching is a residual, right? A loss function like we call it in machine learning. It occurred to me just one day, there's nothing stopping us from making this a lower resolution. In other words, what that paper I just showed you was atom to atom. But in principle, you could project these forces onto coarse-grained sites, that's the alpha, and you ought to get something, right? And my intuition was that will, the Paranello touch, right? It derives back to having had this in place with a simple insight that this could be coarsened, and there you are. And this is now the origin of what I'll call bottom-up coarse graining. To be fair, Scott Schell has also come up with a different approach. We didn't think it was a way to go because it requires iteration, but this is called minimizing relative entropy. Your coarse-grained model, you want to minimize its relative entropy to the all-atom model projected onto the coarse-grained model. So this is called relative entropy minimization. I will say, and then I'll get into going beyond that, is now it turns out that they work very well together. If you use force matching as a start to get you a guess, a good starting point, to then feed into relative entropy minimization, which needs to converge, right? You iterate these you see all the brackets, right? It works beautifully. And so the two methods are complementary, and so we can do that. If you're interested, by the way, there's a review article now a few years old, but still quite pertinent, plus software that's available. Okay, that's not good enough. We wanted to go to larger scales, and we've always wanted to do this. And I've shown this picture many, many times. These are these actin filaments. These are cells. These filaments are in there. They push things around. They move the cell. They do all kinds of stuff in a single filament. And it occurred to me again, insight, I think, comes before, writing down the math. What I've been telling you about isn't going to be good enough. We still have too many coarse-grained particles. We need to coarse-grain these proteins dramatically. We need, to reach these scales, we need these coarse-grained particles to represent a lot of amino acids, not just a handful of atoms. And so this is what we call ultra-coarse. We have internal states, and the internal states can change depending on what the particles are doing. An easy way to understand that is this copper ball is representing the nucleotide. It can either be an ATP, it can have undergone hydrolysis and have the phosphate group cleaved, or the phosphate group can have dissociated. So that ball needs three states. And the kinetics of that depends on what the other balls are doing, and the other balls depend on what it is doing. So it's a coupled quantum-like, or an isomorphic quantum particle. And that allows us to incorporate a huge amount of information I'll call it implicit, in these coarse-grained models. I mentioned hydrolysis, it could be, electron transfer, protonation, ligand binding. All this can be folded into these models if you just do it right. Okay, and this is the last tribute to the Paranello visit. I also had the time in Lugano to actually think. I don't know how many of you, if you're in the United States, you spend most of your time trying to fight off budget cuts or write grants, and it ain't funny. Okay, but I had a little time there in Lugano to think, and my student James was there. Michele's not on this paper, but we were able to write down the first paper, the theory. It's always important to have a real theory behind what you're doing eventually, one way or the other. Okay. So how does it work? You have everything we had before. You just have a set of state-like parameters. You have to figure out two things. How do you identify those states? And how do you then propagate the dynamics? And the identification is not trivial. I'll give you an example. The particles are in local equilibrium as they move, and the states are a linear combination of it, sort of like an Ehrenfest theorem or something like that. So you can write down this, and you can take derivatives. If you will, this is, if you don't sort of understand what I'm talking about, this is kind of a marriage. So a simple example, I won't go into the details of this, but you can ask yourself, how do you do something like an interface? Because the algorithm I told you about, without this ultra-coarse graining idea, you would train on the liquid, and the vast majority of the interactions are in the bulk here, right? There's their interface. And So your coarse-grained model really knows about the bulk. It's been overwhelmed by all this. But if you have a way with an order parameter that tells me, okay, I want to train only when I'm at the interface or in the bulk or possibly in an in-between, you can develop this coarse-grained model that understands you're at the interface or in the bulk. It's kind of like a polarizable force field in some ways, right? But a coarse-grained one. So this is a very powerful application of that. Now, more recently, again, we have to get on the bandwagon here with machine learning everything. And the idea is if you have an idea about what the definition of the states are, right? So I don't know the fancy words, but I guess that's supervised. In other words, I'm telling it that I know there are certain states. There are ways using a machine learning like loss function, which is a little non-trivial really, but an entropy-based thing to train these states. And this paper was just published in Journal of Chemical Physics if you're interested, all right? So this has come a long ways. And it all started in being able to think in 2012 without interruption for a few weeks. And it's a sobering thought that, that's what it takes, because it's hard to find that time. All right, so now I'm going to give you a major application. And in sort of looking at who's likely in this audience, with the exception of Pratouche, most of you have probably not heard what I'm going to talk about. But I've always been easily bored and also liking a challenge. And so I decided, well, with all this coarse-graining technology, we should be able to really get into parts of it. But the key part, or a key part, is that an infected cell sends off a virus particle. It's got these proteins have a symbol. They take the virus RNA with it. And then the HIV protease, an enzyme, starts chewing up this protein called GAG protein. And the green part assembles into the virus capsid around the RNA, right? And this is a deadly delivery object. It comes in, and I'll talk about the second part of this in a bit. It targets a new cell. It goes into the nucleus of the cell and integrates. And so that's how the virus works. Now, it occurred to me, well, we ought to be able to do this if we're worth anything, right? With all this fancy talk and all these equations. It turns out you can self-assemble these capsids in vitro without all this biology. You can do that. Just put the proteins in, put a certain special ion I'm going to talk about in a minute, and you will get these capsids even without the RNA. So you ought to be able to do this with validation, right, from in vitro experiments, right? The full biology is pretty hairy. So the capsid looks like this. It's an interesting cone shape. These proteins dimerize. There's 1,200 of them, monomers, that go into this capsid. You form these hexameric lattices, which are actually non-trivial. 1/2 of a dimer is in one hexamer. The other half is in an adjacent hexamer. And according to Mr. Euler, you need 12 pentameric defects to close this shell, right? So that's what the red, they're not different proteins. There's pentamers rather than hexamers. So we coarse-grain this. And we got some early results. We published a paper, but it was often you wouldn't get a good capsid. Most of the time it would grow from down here, which doesn't seem right because you need to incorporate in the virus the RNA, so you really think you should grow from up here. It turns out we had left out two critical things. One is, of course, that the protein is got a lot of motion, right? You would have known this. We should have known it. In hindsight, it's trivial. This is the C-term, it's very strong. Now, you can't do this explicitly, all right? One simulation of one protein, you try to map it into a Markov state model to study that, it will just eat up everything. It's very hard to go beyond that. But this is where this ultra-coarse-graining idea comes in. We can actually affix a dynamical variable to these proteins, and we can say, yeah, I'm in the right conformation or I'm not in the right conformation. So just a binary variable. It could be more complicated. You could try to blend in more additional conformations. But that deals with the entropy of this protein needing to be able to sample all these things, but it needs to find this right conformation. And you can start assembling. I'm not going to show you those because I need to go on to this. All right? Along comes the structural biologist. They have a way of ruining your day. You know, they have new structures all the time. And, you know, we worked really hard on this problem, I got to tell you. So they come along and say, you know what? There's this anion that we see stuck. It's called inositol hexaphosphate, right? It's a phosphate anion. It's ubiquitous in your cells. And it likes being in these hexamers because there are some arginines, positively charged, that bind it. You can see this is just mutants, don't worry about this. You can see if you don't have IP6, you get tubules. The protein actually likes to form a hexameric lattice, but long tubules look like nanotubes. If you add a little IP6, you start getting cigars on the ends, and there's a few. You add more, and if you look really closely, there's cores all over the place. We call them cores. And so IP6 is important. So what we had to do was go back and calculate the binding of IP6 to hexamers and pentamers, either from the bottom or the top. That's why we come in two directions. And what you find is something counterintuitive. The pentamers like IP6 a bit more than the hexamers by about 5 kilocalories per mole. And the reason for that is the pentameric pores are tighter, so even though you have curvature, right? That's what they're there for. And then IP6 binds to them and stops them from going away, all right? So the pentamers come, IP6 in low concentration binds to them, stabilizes them, and the rest is history. So you see here, we form a shell. If it weren't for the pentamers, it would go to a tubule, right? But you can see, if you look very, very closely, there is a pentamer, there's a pentamer, IP6 is binding to it, and you get the cap, and then it's game over. This thing says, I want to be a tubule, but I can't, right? It's too late. So you have this shell, and you form it over and over and over again. It's completely reproducible with some polymorphism, you call it, that agree precisely with cryo-electron tomography experiments. So it's really a remarkable thing. And it also tells you that these are glass-like structures. They're not thermodynamic minima, like a funnel picture. They come and they get frozen into a certain structure, and then that's it, and they go on. Okay, and this is what you just saw, right? Number of coarse-grained time steps, so this is time. Number of pentamers, you see just very few of them, 2 or 3, and then it explodes in growth up here to over 1,000 proteins and then saturates. And so it's this very interesting behavior. Okay, now let me show you about a second part of this. This is a brand new thing, a couple years old now. People for years thought, well, okay, the virus attacks you, the receptors, it goes into the cytoplasm, it uncoats here, the genetic material finds its way into the nucleus. All right, and then, and I'll show you a picture in a minute, it's amazing. People discovered that, in fact, this capsid is going into the nuclear pore complex, into the nucleus intact, right? So this little delivery agent is taking the deadly RNA of the virus all the way into your nucleus. And here, see this cryo-ET picture? There's the nuclear pore. There's an intact capsid, and there's been multiple other experiments now that have shown this. And do what I'll call an integrative coarse-grained model. You know, it takes some courage to do something like this because it's really complicated. And so we can put all these things together into a coarse-grained model for the nuclear pore complex plus the capsid. There it is here, and there is a simulation of the capsid docking into it. And there's no ATP, GTP. It's an electrostatic ratchet and it may explain why the cone shape is important, right? If you think about it, right? So the cone gets its nose in there, and then it electrostatically ratchets. The full nuclear pore complex has a thing down here called the basket, which we don't have a structure for yet, and it helps shepherd this thing all the way in. And you can start to say things. For example, if the nuclear pore is constricted, they fluctuate, it'll spit out. None of these will work. If it is fluctuated open, if the cone comes in the right way, like you're seeing here, it will dock nicely. If it comes in the wrong way, it will get spit out. This interesting pill-shaped, and these do exist. We actually see them in our simulations. They will go in. They're fragile. There's a lot of curvature, so they break apart. But we can, this paper was published last year. And how does it do this? So it turns out that this capsid is pretty flexible. So these are pictures of compressive, and this is actually from a 100 million atom, all atom simulation. And there are these compressive and expansive domains. It's kind of like a spring like this, ready to compress some, and also the lattice itself will disorder a bit. So entropy goes in the right direction, and enthalpy goes also in the right direction, and this thing can survive the nuclear passage. And you can kind of see that as I took pictures. you can see the compression part of the springs as it goes in. So quickly, let me wrap up. By my, 5 minutes until questions, right? But if you're lucky enough to get it, you may survive. So, anyway, Lenacapavir binds in substoichiometric quantities to the lattice. And it likes sort of distorted regions, so it doesn't need to bind to every capsamere, but it does bind. And so we could put this into the model also. And what it does is makes this capsid like an eggshell. These are stress sort of stabilization domains on top of what I showed you earlier, right? This is on top of that. So it goes and it binds and it makes this into an incredibly rigid object. And what happens to that object, this is a cool movie. You'll see, this is working its way into the nuclear pore. It's a little bit long-winded, but just hang on. You'll see, as it starts experiencing stress, it has Lenacapavir. If you look carefully, you'll see little orange balls. You'll see the pentamers, which are unstable, really. They're there to help stabilize curvature. You'll see them pop and break off, right? Please, it takes a minute. And then you're going to see some remarkable allostery for lack. See that translated all the way up to here, these pentamers. Now you start seeing a seam break. Okay, it starts tearing. And this simulation does not have the contents, the RNA contents. We have simulations of that, and it starts, coming out, as you'd imagine. So, what does Lenacapavir do? It rigidifies the capsid and does not let it get through. This is with RNA. Does not let it get through the nuclear pore. That's the last line of defense. If Lenacapavir failed here, it does multiple things. It sort of stops capsids from forming. It may even in the early stages do things, but if it doesn't work here, then you're in trouble, all right? And it does. And then, so the question is, can you really believe any of this, right? I mean, this is where the courage parts come. This is you're trying to get real virologists and molecular biologists and, backward-thinking other people that don't want to believe you. You try to get to, and so you better do some experiments. Well, it turns out, there's another experiment in our manuscript which has been submitted where you label the capsid and the contents, and this is the nucleus, and you can see that it goes away here, right? This is this as they break, the RNA goes away. Okay, so I'm going to mention one thing. You're just going to have to give me that. So I have a bunch of stuff that's under development, but one of the most important things I think where the field needs to go is backmapping. And we just published this paper. So the idea is you can have all kinds of coarse-grained simulations. Hopefully, you believe in them, they're accurate. But in the end, we really do need to be going backward to the all-atom representations, right? To understand the validity of what we predicted, to maybe iterate, improve what we're doing. And this uses these diffusion maps, right, from AI technology to go from coarse-grained representations. And it's remarkably powerful. For example, we can backmap the whole capsid, and we can even backmap the IP6 in it. And so this is very powerful. And then I guess I'll end up here. This is another thing we've been working on is blending quantum mechanics with coarse graining. So, the cost of any of these simulations is the QM, right? It takes a lot of time when you have to do QM. So we've figured out how to blend that with a coarse grain representation, which saves you a lot. Let me quickly, see, I had lots of stuff to talk about here, but go to the end. I want to give credit where credit's due. These are the people who have done the work. Many of them have moved on to new things, and it was the funding, so we'll see where it lives. And that's it. Thank you very much.
Thank you. Thank you very much, Greg, for the inspiring and great talk. We have time for questions.
So I haven't understood how you perform
Which of those states are which? And I'm not sure that's in I think it's in our software now. But that's on top of what we started in 2004. Yeah, that's the so-called ultra-coarse-graining. Yeah.
You haven't discussed that?
Yeah, we have. It's published in a number of papers. And I think I did.
Just
quickly.
flash.
I mean, there's always pretty heavy theory stuff behind it, but I don't think I got time to do that. But this in Journal of Chemical Physics, Patrick Sharman is the first author, and I think it's this year. Yeah.
Greg, Greg, you mentioned how backmapping is important, these kind of things.
Yeah,
We just
that's
recently
what I
published. Yeah, we just recently published a machine learning stuff on that. But I was just wondering also about the mapping, because you show, particularly for the Capsid, how important it is to have an atomistic sampling that, from which you can map the ultra-coarse-graining. Could you please mention?
You mean backward or forward?
The first part, so mapping before doing the ultra-coarse-graining simulations.
Well, in this paper we've published, we are able to do quite highly coarse-grained models, and that then will feed into something that is less coarse-grained, and your software I know about, but also there's others, right? And so at some point when you've done that backmapping, you want to equilibrate your all-atom structure, right? Because that's important. And there's no free lunch around doing that. Yeah, yeah.
There is the time for the very last question, if there is for Greg. If not, so let me thank again Greg for the nice talk, and we can move on with the program. The next speaker. Thank you very much. So, next speaker is Dhiman Ray, and he's going to talk about integrating a bias path sampling with bias enhanced sampling for rare event kinetics. Thank you.
Hello. Yeah. Okay. Thank you very much for inviting me to this conference to celebrate Michele's 80th birthday. So today I am going to talk about a little bit about my work in the past 1 year. But before that, I just want to thank Michele for the memories that I had from, because I am one of the most recent alumni from Michele's lab, and so I left the group in like exactly 1 year before, and that was the group at that time, and we had a lot of fun in Genoa, going to the almost every other week. And the last picture that you see on the left, that was like his 79th birthday, which was coinciding with my last day in Genoa. So we celebrated that together. And going from Genoa, I actually moved to the States at the University of Oregon in the city of Eugene. So those who were asking me yesterday, it's about there on the Pacific Northwest, in the just little bit up of California. Variables using machine learning. And on the application side of my group is focusing on RNA ligand binding and protein-protein binding. And for this talk, I will focus on this integrating enhanced sampling and path sampling methods for getting kinetics, because we see a lot of people are talking about machine learning, so I'll skip the machine learning from my talk. And so this audience doesn't need an introduction on rare events, but what we are concerned about here is we are trying to calculate the kinetics of rare events and not just the free energy landscapes. And many of the methods that have been developed in the enhanced sampling community focuses on the free energy landscape itself, but the kinetics is also important. And that's because in our human body, all the cells are like open systems. So there is a flow of materials inside and outside of the cell. And so none of these cellular events take place in an equilibrium environment. So for that reason, the rate constants are really important in the biology. And sometimes people say that biology is all about kinetics. And some experimental evidence for that is also visible. So when we people have looked at how the efficacy of a drug actually related to the binding free energy and kinetics, they found that the kinetics is better correlated with the in vivo drug efficacy. But the problem with enhanced sampling simulation is that it's getting kinetic properties. So in 1995, the year I was born, which is exactly on the 50th birthday of Michele, so this new method was introduced, which is the conformational flooding, where Helmut Grubmüller has shown that if you could apply the bias lesser than the actual barrier height, and if you do not apply bias in the transition state, you could actually get kinetics out of in bias simulations. Now, how is this done? This is done based on the transition state theory, and where it can be shown that if which are related to the transition state, like the partition function of the transition state or the transmission coefficient, those things will remain unaffected. And with that assumption, we can write a ratio of the biased time scale and the unbiased time scale in terms of an ensemble average of the exponential of the bias deposited in the initial state minimum. Now, this technique has been translated by Michele and Pratyush, who is also in the audience, to this infrequent metadynamics algorithm, where they are depositing bias with a very larger pace, which is the time interval between the bias, the Gaussian depositions. And with the way they are expecting that because the transition state is short-lived, so there will be no bias deposited in the transition state. And there is a large body of literature in biology, material science, and chemistry on using infrequent metadynamics to get kinetics. And me and Michele wrote a review when I was a postdoc in his group. And that review, I planned to put like some examples of where this has been used. And it in the Journal of Chemical Theory Computation, it fills 5 pages of table, just the applications of infrequent metadynamics. And I just copied like 3 pages, which responds to ligand binding, other biological problems, and chemistry and material problems. Now, there are still, although it has so much application, there are still some limitations to this approach, because we are assuming that the barrier is a very sharp barrier and it's very short-lived. But in case the barrier is more diffused, it is less likely that we can completely avoid any bias deposition in the barrier. And number 2 is that because we are depositing bias at a very longer interval, so the growth of the bias is very, very slow. And so it will take much longer time to see transitions than we could see in regular metadynamics. We also have an excluded region parameter, so using which we can explicitly tell to which parts of the free energy landscape, for example, where the transition states are, to not to bias. So we can explicitly avoid biasing the transition state. So this is a method which improves over well-tempered metadynamics by giving us more control over where to bias and how much to bias. But when I looked at more complicated processes. So in biological systems, they involve both barrier crossing and diffusion. And this type of system, for example, like ligand binding, where there is a very deep free energy minima at the binding pocket, but once the ligand comes out of the binding pocket, it has to diffuse through the protein surface to actually go to the unbound state. And in this process, it is still interacting with the protein. So that is why the free energy landscape will have some shallow minima and a rugged free energy profile. On that, it has to diffuse until it goes completely unbound state. And this problem is not only specific to proteins, but it can be also seen like multi-step nucleation processes, and also in case of protein folding, where we will have a rugged free energy landscape, where you have to do both barrier crossing and diffusion across the landscape to go from one state to another. Now, in most of these problems, what we usually have for starting our simulation is only the structure of the deepest minimum, which is usually the bound state in case of a ligand or maybe the folded conformation of a protein. We do not know how the rest of the free energy landscape looks like. So it can be difficult to actually set up a system to do opaque flooding to get transitions across all these high and as well as smaller barriers. And here is an example why that is a problem. Suppose we know only about the deepest free energy minimum, and then we developed an opaque flooding protocol which will bias in the minimum and then not bias in the rest of the free energy landscape. And I am showing the free energy landscape, but you need to think that we do not know it at the beginning when we are setting up the simulation. Now, what is going to happen to seconds, and that is difficult for us to sample using unbiased simulation. What we can do is we can try to do opaque flooding for each of the minima that is present. But to do that, first of all, it's going to be computationally expensive, because we need to compute the full free energy landscape to know where exactly all the minimas are located. And that is computationally expensive, plus also in some cases, we just want to get the kinetics. We do not want to explore the full free energy landscape. And so, how can we go about solving this problem? So, before going that, so let's just summarize what are the issues that we are talking about. So, in OPES flooding, it is very good at sampling the barrier crossing processes. These are fast, but the diffusion process is slightly slow. And so that we can get the kinetics, but sometimes it can be difficult. Now, so here I will introduce another method called weighted ensemble. This is a path sampling algorithm which I have worked on during my PhD. And this method is actually done in a very different way. So here we do not apply any bias, but still increase the sampling by trajectory resampling approach. So here, for example, you are in the initial state, which is the right-hand bottom corner, and then from there you start sampling. And when we divide the whole conformational landscape in a bunch of grids, and then when the system moves away from and reaches a new bin, we split it into multiple trajectories. So merge some of these trajectories to make sure that the total probability is 1, and the number of trajectories per bin is the same. And we also resample the weights. So basically, at first, this example starts with 3 trajectories of weight 1/3, but when it goes into other bins and then splits into other trajectories, the weights decreases based on, and the weight is redistributed among the child trajectories, transition time. Now, this one is also very good, but this is not OPES flooding. And this does not have an explicit mechanism to push the system over the high energy barriers. It can do it, but we have to do many rounds of resampling, and it becomes extremely expensive. So, but it is very good at exploring rugged free energy landscape, which is filled with short minima. And so the sampling gets much better in that shallow region. So it can also give you kinetics in certain places it can't. So what our solution was to combine these two and make something called an integrated sampling, which is a combination of enhanced sampling and path sampling. So what the integrated sampling is expected to do is to perform this weighted ensemble and OPES flooding simultaneously, so that we can get the benefit of both. So we can also we have a fast barrier crossing and also have a fast diffusion. Now, we can do that, but we need to have a statistical way to get the kinetics correct. And for doing that, what we are going to do is, once we see a bunch of transitions, then we will use the reweighting scheme of OPES flooding to get the time scale of this transition. But how do we define the transition? So we trace back the final transiting trajectory back into the initial state. So here this dotted line actually shows one such trace of a successful trajectory. And then we perform this reweighting on top of that. So we will know what time it would take for that trajectory to actually go to the final state. Now, each of these some weight, and they are not equivalent to each other. So we have to compute an weighted average of these times to expect to obtain the average mean first passage time. Now, we tested it at first on everybody's favorite example, the alanine dipeptide. And here we could obtain a converged kinetics in a reasonable amount of time. So compared to OPES flooding, this integrated sample, look at the free energy landscape. The process that we are trying to study is this transition. So this transition is a single barrier transition. So there is no need for long diffusion across a rugged free energy landscape. So that is why there is no extra benefit gained by integrating the weighted ensemble on top of the OPES flooding. In fact, it takes a bit longer time because we have to do many rounds of resampling. So where can we benefit from this method? Where the free energy landscape is not like a single barrier. So let's look at this Chignoline unfolding problem. And so here the barrier is not just one peak, but it's a little bit rugged barrier. And here we are seeing that what is the benefit of using integrated sampling. So the orange trajectory is actually an OPES flooding trajectory, where we can see the system is coming out of the folded state, but again coming back and fluctuating and spending a lot of time in the transition region before reaching the unfolded state. Why is that? Because there is no bias in that region, so there is nothing to drive the simulation towards the unfolded state. But in case of integrated sampling, once the system comes out of the unfolded state, then it very quickly goes into the unfolded state, because weighted ensemble is accelerating the dynamics that opaque flooding could not accelerate. And so here also we could get converged kinetics in agreement with the unbiased reference. And here we can see that the result of the integrated sampling is Research Corporation. Opaque flooding works well, but we can now reduce the total computational cost because we are not spending the time doing unbiased sampling in a rugged free energy landscape. Now, in case of infrequent metadynamics, the simulation time was even lower, but the overall agreement is not as good as opaque flooding or integrated sampling, and that is for the reason that here the barrier is not a sharp barrier. So there are more bumps in the barrier ligand. Now, what in our case, we ignored the role of water. So we can check that how well we can capture a situation where the full CV space is not known beforehand. So the collective variable is not that optimal. So here, if we project the free energy landscape along the distance from the binding pocket to the outside, we can see that there is a deep minima, and there is also another shallow minima, which is a clear bottleneck, and finally reaching an unbound state. Now, I am showing this picture, but you need to remember that when we are looking at to get kinetics, we do not have this free energy landscape in the beginning in our hand. So that is why we only know about some conformations in the bound state. So what we usually do is that in this sort of situation, and what we did here as well, we perform an unbiased simulation in the bound state and see which region of our CV space it is exploring, and we deposit bias only in that region. And the rest of the thing is taken care of by weighted ensemble. And when we do an integrated sampling like that, within about 200 iterations, or even we can say below 100 iterations, we can get a converged estimate of the residence time, which is the ligand unbinding kinetics. And if you compare the time scales it took to converge the opaque flooding and integrated sampling, here we can see the benefit kind of becomes bigger because we have a huge bottleneck in the middle, which the opaque flooding cannot sample because we do not have the idea of the full free energy landscape to start with. So we can also show that even though we did not include the water collective variable in our CV space, we can still some really realistic applications of proteins and things which are medically relevant. And my postdoc Shompriyo has looked at this open to closed conformational transition in heat shock protein 90, where this open conformation can bind a drug. But in this closed conformation, which is reasonably close to the experimental NMR relaxation studies, which is a factor of 6 or 7 away, which is kind of acceptable considering the limitations of the force field. But within the same or similar amount of opaque flooding simulations, we were unable to observe any transition because the ruggedness of the free energy landscape. Now, with that, I would like to conclude saying that what are the main messages. So, we have a method which combines the biased enhanced sampling and unbiased path sampling to efficiently calculate kinetics, and the choice of CV is not very critical, and it can drive transitions along the minimum free energy pathway. Now, we have made an open source implementation using the Plumed and the WestPath software, which uses enhanced, weighted ensemble simulations. And I was a part of the developer group when I was doing my PhD. And in Plumed Nest, the input files are available for everybody to try and test it. And in the future, we want to use this technique to explore the landscape more and be able to compute even the free energy landscape in a more efficiently than regular OPACE. And one of the things which is challenging here is to solve the trajectory correlation problem, because here the trajectories are splitting and merging. So all the transitions that we are seeing at the end are not independent. They share some part of their history. That is why, although we see a very high number of transitions, we cannot actually do a fitting or a Kolmogorov-Smirnov test to see that whether our kinetics is reliable. We cannot do that because these are not independent variables. So there are correlation between this data. And to solve that, we are currently putting some effort, and also other people in the weighted ensemble community. With that, I would like to thank my group members and University of Oregon for the funding, and I will be happy to take any questions.
Thank you very much, also for being on time and for the interesting results. We have time for questions. Is there anybody from the audience?
Our work is good for 1 and 2 dimensional mostly, But going beyond that, even for creating the weighted ensemble grids, it becomes exponentially higher number of grids in very high dimension. So up to 1 or 2 dimension is something that we kind of do with this method.
Thanks, Dhiman. Very nice talk. So I know you mentioned it in your future directions, how to test the quality of the kinetics, but I wanted your general thoughts on how you would approach it, particularly for weighted ensemble. The story over the last few years have been that it can show false signs of convergence, but as you do more runs, if you keep running it longer, the flux can suddenly drop down very much. Are you not facing those type of issues? And maybe you're avoiding them because you're not doing weighted ensemble for the deep part, you're doing it only for the diffusive part. And secondly, connected to that, are you sure that the choice of CV is not critical? Because even when you're trying to divide the region between where you want to switch from metadynamics to weighted ensemble, you probably still need to know what is a collective variable that distinguishes enthalpic parts from entropic parts, right? So you could only benefit from a good CV.
Yes, I will answer the second question first. So we indeed get a benefit from good CV, and the point is that even we do not have the best CV, still we can get some like an accuracy if we run reasonably longer, but in case of OPS we might get completely stuck, and that is one of the issue. And about the first question, which you were mentioning about the sudden increase of flux, and that is actually true. And that is the reason why I am not using the flux to actually measure the kinetics. So I am using the weighted average. And why is this working? Because this is an approximation. This is not the exact solution of the kinetics. But the majority bottleneck of the kinetics is coming from the first barrier crossing event. So that is being taken care of by the OPS flooding. And what we have tried and we have seen that at the end, if we use, when we have like a fully converged system with like thousands of transition, there whatever method you use at the end to incorporate the average, it doesn't really matter much in terms of the kinetics, because what is the main part?
Completely ignored the kinetics calculated from the weighted ensemble, your answer would still really depend only on the rate constant you are getting from the OPS part. The weighted ensemble is not going to add much to the time scale if you completely ignore it.
Is not adding, that is because it's a, this is the flooding free, like the flooding kinetics that we are taking the weighted average of, right? So because if we are biasing, and you might have seen this also before, that in the OPS flooding that the 99% of the kinetics is for that part where it is biased. Once it comes out of the bias, it adds very little. And so that is going to happen.
A follow-up question at this point. You clearly mentioned the fact that you're using the weighted ensemble for the semi-diffusive part of your Fourier surface. I'm just wondering, on the same line, how important is the binning that you define when you perform your weighted ensemble? Because this may also, if you have to cross very low barriers, this may...
Yes, and that's a very important question. In older days of weighted ensemble, when I was as a PhD student, we had to manually do the binning and pick like where the bins are. But now we have an automated method which can pick the bins based on the need. So in the regions there is less sample, it will automatically split. So the binning I shown in the picture is more like for visualization, but the actual binning is automated and they're adaptive. They are changed based on which part of the free energy landscape, what is the gradient, and all these matters. So user doesn't need to define the binning anymore.
Okay, is there any additional question? If not the case, let me thank again. And the title of his talk is Molecular Transport Phenomena with metadynamics and beyond. Thanks, Matteo.
Okay, thanks to the organizing for preparing this nice meeting. I'm really honored to speak today. And in fact, I'm part of the first group of people that moved. Some moved from Stuttgart to Lugano. This is taken at the rooftop of CSCS, probably in this period in CSCS. And my interest is transport phenomena in confined system, in particular the nanopores. And in fact, I started this topic when I was with Michele as a side project. In fact, we applied it for the first time, the metadynamics, to the transport of an antibiotic. So why nanopores are important? For sure, for nanotechnological applications, there are devices that can read DNA, and now many people working for also sequencing the protein using nanopore. They are important for early drug discovery because we know, for example, in bacteria, the main path for antibiotics to enter the bacterial cell are porins. And porins are used not only for DNA sequencing as a biomimetic system, but also now there is a huge interest for sensing small analytes. So that's the outline of my talk. I will talk about diffusive transport through nanopores, the concept of permeability with machine learning, and the application to early drug discovery. So the problem in gram-negative bacteria is that we don't know how to measure the permeability of molecules, but we have also receptors like siderophore transporters, and the molecule can also go through direct diffusion, but the outer membrane is very thick. So in fact, in the end, the simple porins are the main path to enter. And recently working with a colleague from biology in Cagliari that is interested in bacteria living in extreme conditions, we found that bacteria can have a third layer that is called the S layer, and also in this case there are porins that we are investigating. So the model used for investigating porin is the OMPF, that was the first protein to be crystallized in the 90s. And the shape is as an hourglass shape, so we have a site constriction in the center and the so-called charge segregation in the center. So on one side we have positive residues, on the other negative residue. And if you look at the formula for the permeability, when we work at a small gradient concentration, we can use the fixed formula. So the problem is to calculate or to measure P, the permeability coefficient. And the permeability coefficient, if you look at the 19th century, depends on an integral over the free energy divided the diffusion constant. And in fact, the flux depends of an integral inside the pore of energy, so statistical descriptions. So for example, docking does not help to identify good substrates because it lacks a statistical description and it is a local method. Instead, we need to know all the interaction of the molecule inside the pore. So if you look at the steric effect because of the hourglass shape, in fact, we were really surprised when we started to see at the dimension of the pore and the dimension of the molecule. We see that if you take standard deviation, and the molecule can go through because there is a small overlap along the tail of the two distributions. And the question is standard MD simulation enough to investigate the distribution of the internal sites for porins? No, in fact, we proved recently that if you use parallel tempering, in fact, we can see also the gating that with standard MD simulation we do not see. So sometimes this pore spontaneously closes. And more recently, we sent the paper to JCP for the special issue. We ask another question: what the size of a molecule is? It's a simple question. So we took a very simple potential, a clepsydra potential, and the molecule can go through. So we calculated the free energy for a few molecules to go through at different internal radius of this clepsydra potential. And we calculated with metadynamics the free energy. Here on the right we have a comparison of metadynamics and standard MD simulation for the free energy, the barrier. And we see if the pore is quite large, they are good, but otherwise we have to use metadynamics. And so we calculated for 5 antibiotics, and we tried to fit the free energy barrier using a 2-parameter Gaussian model, so a radius and a standard deviation. And we see, for example, that for tazobactam, that is the smallest molecule we had, and the barrier is to the fact that the molecule has a very low standard deviation, so it does not fluctuate. Instead, the other that has a bit larger, but with a large standard deviation, they adapt better when the pore becomes smaller. And there is an important point because of the distribution of the three principal moments of inertia. And for example, we saw that for a molecule 1 and 2, our Gaussian radius is exactly the middle, the principal moment of inertia 2, but not for the other three molecules. And the reason is that because of the third, the largest principal moment of inertia, that in these other cases is far from the second, so when the molecule is inside the pore, a small rotation of the molecule has an effect on the barrier. So in some cases, we need to consider all the principal moment of inertia. So, because inside the pore we have the so-called charge segregation, we investigated the electrostatic effect, and some years ago, in fact, we arrived to evaluate the electric field inside the pore. The idea is simple. We took a box of water, we applied an external electric field, and we calculated the polarization of water as a function of the applied electric field. And our simulation is in good agreement with the Booth formula. That is an empirical formula. And then we did the opposite for porins. So we took, because our water-filled pore, we took some the water molecule inside the porins, and we calculated the polarizability. And from this plot, we obtained the direction and strength of the internal electric field. It is a transversal electric field in the center of the pore with a strength of 35 millivolt per angstrom. It is quite a large electric field. And so the question is how we can demonstrate that this internal electric field exists. So we asked our colleagues in Germany that they perform electrophysiology to do some experiments. We took norfloxacin as a probe, and norfloxacin is a dipole energy. But in electrophysiology, you apply an external electric field that is longitudinal, so you help more or less the molecule to rotate. So we expect that varying the external electric field, you can have more events of translocations. So we expect an exponentially dependence from the voltage. And in fact, when they measured the events, they found that the number of events depends exponentially on the voltage. In fact, this behavior is not reproducible by electroosmotic flow, by the movement of water due to the selectivity of the pore, but is in fact due to the interaction of the electric field with the dipole of the molecule. And another point that was important is that when we have the cis addition, the molecule is added to the cis side, we have events with both the electric field, positive and negative. Instead, when we add to the other side, to the trans side, we have event only at positive voltages. But we can even change with mutations, so we can make the system more asymmetric with mutations, and to have a system, a pore with only passage with negative voltage upon cis addition, passage with positive voltage upon trans addition, or an homogeneous case. So the rule is that molecules associate with the positive group in assassin with a low external electric field and with high external electric field, and we see a difference at high external electric field. The molecule is tilted, and so it can go through with the lower barrier, so we have more positively with the transversal electric field of the pore, and we have a compensation of the central barrier. In fact, the binding that is measured experimentally, in fact, is not necessary to calculate the flux of molecule, but is useful just to be able to measure the transport. In fact, we revealed fast events with the residence time in the sub-microsecond range, so molecules going through in 500 nanoseconds, that is really close to the diffusion limit. Okay, but the calculation of free energy is really tough. I mean, we need hundreds of microsecond simulations. So we try to find an approximate way to evaluate the permeability coefficients. And because of the shape of our free energy, and that is in fact is very high in the center, we can approximate the integral like with the highest value of the barrier. And there is the formula. And experimentally, in fact, they measure the logarithm of P, that in fact nothing else but the energy. And the energy, the interaction of the molecule with the pore, can be divided, we said, in two terms, a steric part and the electrostatic part. So it means dipole with electric field and charge with the internal electrostatic potential, plus a constant. So we measured for 80 molecules, and using the property of the pore and the molecules, we obtained the alpha, beta, gamma, and delta parameters to obtain, to fit the permeability. So in this way, we can predict, once we know a new molecule, a new pore, what is the permeability. And there is the plot of the correlation between our data and the predicted data. And but the one critics to this paper to predict the permeability with two objectives. One, to have an interpretable model, so with small number of descriptors, also to avoid overfitting, because in fact, in the end, we don't have a huge number of data points to use. And then physics-based, we want to keep the physics description to consider the steric effect, the electrostatic effect. So we analyzed the data from Paul Ergenrotter, that is a chemist in Urbana-Champagne. He had a few hundred molecules for which he measured the accumulation that is easier to measure than the permeability, but they are correlated. And so we took 15 molecular descriptors for the small molecules, and we used all possible combination between one descriptor and 15 descriptors. And in the end, the best results for a model predicting the accumulation is a model with 5 descriptors: the volume, plan of best fit, the charge, and the PMI1 and PMI3. And that's are the results. But we know that, for example, for some descriptors, it is important not only the average value, but also the standard deviation and the detail. So we added the 3 other statistical descriptors, 5 additional descriptors. We improved the physical-based descriptor. We can improve even more the predictions. And so in the end, now we are a few models by which for this bacterium. And we calculated the permeability. We measure the permeability of other molecules, not antibiotics, and we got the machine learning model for predicting the permeability. And then in the end, we had the correlation between 40 data from MIC and some data from measured permeability and the other from predicted permeability. And we see that, in fact, there is an anti-correlation between the permeability and the MIC. So when you have a high permeability, your molecule has a low MIC. And another important point is that at low permeability, you have a large variation in the MIC. So probably when there are just a few molecules that can enter, there are other effects that can stop the molecule to inhibit the bacteria. So that's the end. We restarted from our physics-based concept for permeation of small molecules, so that uses the compensation of the steric free energy barrier with electrostatic. Now we focus only on small molecules because the idea is to screen very large databases. And we explored the interpretable and physics-based model to predict accumulation for the moment in Escherichia coli. And since the molecular descriptors are not distributed as Gaussians, we introduced the descriptors that can characterize the descriptors, so the standard deviation, the kurtosis, and the skewness. And the accuracy and precision are increased when we add this statistic descriptor. So I know that there are people working with hundreds of descriptors. We use just a few with a good statistical descriptor, and we obtained better results. So thanks to the folks in Cagliari and around, especially Stefan Milenkovic for the Forte Island, that is an island in the southwest of Cagliari, with a sailboat, an historical sailboat is called Vela Latina. There are just a few cities where they still compete with this boat, and we had the pleasure to go around the island and to enjoy of the sea of Calafossa. So happy birthday, Michele, and thanks again to everybody.
Thank you very much, Matteo. Beautiful talk and great pictures, a great sea. We have time for questions. Probably I can start with, I have a couple actually, but I'll start just one. We may say that the graph that you showed about the decrease of the Messi with respect to permeability may be somehow related to one of the known resistant mechanisms of bacteria towards the antibiotic, which is the pump to reflux. So it could be something that is predicted by your model.
Okay, in this case, probably we took Escherichia coli without the efflux pump. So this is just to evaluate the effect of the permeability and the
since
the target,
it's correlated.
Sure. Of course. The question is that the efflux pump is a method to resist that is very powerful, but it consumes a lot of energy, and is not always active. So at the beginning, you have bacteria without efflux pumps. Then you can inhibit the growth, but if you continue to use antibiotic, then they start to express efflux pumps. So we think that if you have molecules with a good permeability, probably you arrive fast to inhibit the bacteria and to produce, avoid to produce the efflux system.
Yeah, and then you have to combine with other antibiotics.
But of course,
perpendicular, right?
Yeah.
I would expect it to be the square in the electric field. It's electrostriction, right? Did you see that?
The square?
Everything should be a function of the square of the electric field. What I naively would expect.
Okay, okay, in the scoring function.
Yeah.
Okay, this was the first version, and now we have other version where we use a different functional form. And if I remember well, there is somewhere the square of the.
It's electrostiction. The Benedetti has a nice paper about this. Have a look. It's already rather old.
I'll check. Thanks.
Other question? We have still some time. So if not, I have another one.
Yeah, sure.
Since I was very interesting in the part when you measure with the simulations the permeation of the small molecules.
Yeah.
Based on my experience, a lot of difference is made by the force field and the torsion parameters of the small molecules. There are, if you use the standard one from GAF or...
I use it GAF.
You use standard one, you don't know, do any quantum mechanical parameterization of torsion?
Not
just
yet,
because
not
the
yet.
steric hindrance is an issue, it's a key.
Yeah, the idea will be, in fact, now to use this new possibility to run simulation at quantum level with machine learning. But okay, at the moment we have just the classical.
Also in terms of electrostatic, because you also highlight static is important, you are also using any kind of, polarizability? No, just standard one.
Yeah.
Okay. So if there is no additional question, let us thank all the speakers of this morning session. Now we have the coffee break. Ali, how long is that? is Valerio Rizzi at Université de Genève. And the title of his talk is The Unseen Architect, How Water Shapes Biomolecular Processes. Wow, please.
Thanks, Vittorio. So it's a pleasure and an honor to be here, thanks to the organizers. And I will be speaking about water and water-aware CVs. And in the picture, I put actually a picture of Trieste, the main square, wet, it will rain very soon, how it shapes. So the landscape, and I was here just 3 years ago, and it was a very good time, thanks to Ali. So, my talk will be more or less divided into 2 parts, where I will be speaking about 2 articles that we recently submitted, one was accepted and so on, and they are about 2 applications, 2 biophysical applications. One is on ligand binding and one is on protein folding. And in both, water plays a relevant, possibly unexpected role, and we will go more into that. So the first one is titled All You Need Is Water, and it's by Mark and other co-authors here, and we sent it actually to the JCP Festschrift. We are part of the wave that Michele Ceriotti said of the last day spike. So it's available on bioRxiv. We are about to send the reviews in. But before going into the details of what we do there, I take a little trip down memory lane as well. I guess this has not been shown yet. It's another birthday. Apparently time goes in birthdays of Michele. This is 5 years ago. Difficult time. It was between waves of COVID. This, I think, is the last, one of the last snapshots of the Lugano group. We were about to move to Genova. And yeah, it was a bit of a somber birthday, but well, many familiar faces are still here, and happy memories from the time. But even if the time was complicated, it was very rich, I would say, in ideas and many interesting. So to mix with machine learning different descriptors. And we were applying it, of course, on the starting systems, alanine and some reaction. So in parallel, okay, I make just one slide for the very few that are not aware about the difficulty of having optimal CVs and possibly the role of water. These are 2 pictures from 2 very good reviews from the past. The problem of enhanced sampling and CVs is that there are hidden degrees of freedom. So if you have only CV1 in this plot, you're not aware of CV2 that not only might not distinguish between states, but even worse, could be problematic in the transition state region. And that slows down terribly any enhanced sampling. So how do you manage? I mean, you need to find CVs and so on, but again, it comes at the detriment of the generalizability of your method. If you study for years the same system, how can you apply it on many systems? It's a very old and difficult problem. And often I argue, and we will see later, that water plays a role, because often it's there in any biophysical problem in solution is there. It's kind of ignored. It's orthogonal to the standard CVs, but it plays a role. So often it is the CV2, the proverbial CV2 that I show in these plots. So again, in the memory trip, very important paper that will come back, so don't forget, by Sergio in the audience. A paper where they developed a fingerprint, so to measure, let's say, hydrophilicity and hydrophobicity of different small molecules, amino acids. It's based on the GOVAR, and they apply it on a small host gas system. So I was there. That played a part also in my next application. I was looking for a more complex application for deep LDA. We wanted to try something bio. Dhiman showed actually the same picture before. We were trying some host gas system and few water coordination along the axis of the host and a few hand-picked atoms of the ligands, often geometrically hand-picked or next to charges, because why not? We will come to that later. We biased that together with the standard funnel distance, and results were much better. Trouble is that, okay, now in hindsight we know, of course, it's a bit expensive. It takes a while. You need to train one for each system. It's not really generalizable, and there is an empirical choice, so not easy. In fact, very fast, later we tried to apply just immediately with Nerges here in the audience to trypsin, and we thought it would be relatively straightforward. It was not. So larger system is not considered difficult. It is difficult. It took a while. we were able to make it, but we had to change things and developed many good ideas by Nerges. So that, I guess goes to the adage of more is different from Anderson and all. Bigger is different, it's not trivial, okay, it works for this larger system, it will work, no. So that was an eye-opener. Plus other good work with the group of Marco De Vivo and Paolo Carloni, so many water-focused papers from the time. Then my time to leave the group of Michele came, and I joined Francesco Gervasio's group in Geneva. And generalizability there was even more fundamental because we're in the School of Pharmacy, so applying and wasting years on one ligand is not really feasible. If you are able to sample that well in a rather general way, maybe you can even assess which force field is more suitable for your problem at hand. So on the sampling part, soon it's relatively relaxed and there is only one bias, and as you go up the ladder of replicas, you get extra delicate, I would say, biases on different CVs, on for example, the multi-thermal, as well to help yourself to better converge without refining to death your CV. So that's the main thing. Of course, it's more expensive. Usually we run 8 replicas. In the age of GPUs, it's not a big problem now to run parallel, but that's that. And to be honest, also for CV developing, I would not use it. In fact, I will show you non-one-opus result immediately, because if it's a bit cheating. If you want to develop a good CV, you shouldn't help yourself with this. If you are into another track, which is the second example, yes, but to develop a CV, I would say it's better to go back to the standard opus. So a couple of words on another recent paper by a PhD student in our group, where we took, again, all these host-guest systems, many, actually 2 hosts and many guests, applied one opus in a general way. We didn't optimize. So we bias a bit of water, but without looking much. We put it into a general strategy where also there is an automated strategy to build the force field, to build the TPR from the force field, and we could assess, for instance, we could converge very stably starting from different binding poses, which maybe seems a trivial point, but is not at all. Then with the relatively large ligands, we tried starting from the given, in the challenge, sample challenge, given pose and some other, not even a pose, starting from out of the pocket, and we found very good agreement in correlation between results from one to the other. But the main result of the paper was actually, okay, it's a bit small, but we could scan literally the force field space in a limited way, but there is no limit, in fact, one could scan even more. Very small, so in a sense we put the sampling quality aside, because that has been, in a brute force way, admittedly, has been solved here. So that finally links us to the article that I wanted to speak about, and a bit of history of it. We were doing the advanced modeling course, and we had 2 very bright students. One is in the audience, Mark, and the other, Tatiana. And normally we would teach the basics of MD, but with them, they were very good. We tried a bit of enhanced sampling, and of course we tried host guests. And they had a thought-provoking, let's say, idea. Why don't we, you say that water is important, but you try that mixing with distance and all. Why don't we bias only water coordination? And we figure out even a criterion about it. And at first I said, no, it's not going to work, right? But it did. In fact, I speak about it now. Then we refined and we developed a strategy. So the idea, again, we go back to the paper that I mentioned earlier of Sergio and Pablo, where this fingerprint, very intuitive way, it's an integral of the G of R. This, again, goes back to the intuition of Michele and his group. And, you know, from that, applied it to a set of ligands, similar system, host guest, than the one that I showed you before. You see, it varies continuously. On the left is just the standard chemical picture, and on the right, colored, there is this fingerprint. So on the red, we call it anti-bulk, more hydrophobic in a sense, in a wide way, and on the blue scale, it's more bulk-like, water around those. So usually charged atoms, of course, tend to be more bulk-like fingerprint-wise. carbon the other way around, but not necessarily. So in a very automated way, you just put a ligand in a water box, you can calculate it, and we provide the script. So a bit technical slide, we all use those, and then the center of the host as well, that is the center of the hydrophobic pocket. We bias it, we put the funnel of Professor Limongelli here, and then bias with Opus Explore, 3 CVs. Opus Explore is very suited, of course, with multi-CV, more than 2, so it's quite effective. Put a standard barrier, slow pace, and go forward. The results are this. You see, the intuition, I would say, maybe my failed intuition, I would say, is that the bulk-like fingerprint would be better. You're around the charge, right? Maybe water sticks. You see, we look at short times, and you will see why. It's a bit hit and miss. After 25 nanoseconds, not so good. 100 nanoseconds better, but still not amazing. The anti-bulk strategy, much better. It was extremely surprising. At 25, it's already pretty decent. At 100 nanoseconds, single replica, independent replicas to get the error bar, is spot on. And it's only biasing water coordination, which is really surprising. This was, so under we see some R squared, and so on. So it's really converged. Later, longer term simulations, but that was surprising. So much that we were comparing, for fun in a sense, the other way, the other side of the coin, let's say. Just geometric CVs, because why not? Of course, the truth stands in the middle. A hybrid strategy, especially in larger systems, would be more effective. But here we are trying to test the extreme to see which CV is more fundamental, more aligned to the slow degrees of freedom. And again, the anti-bulk choice for these small systems, again, seems to be better, which, again, was surprising. Again, many tests show that it's pretty well converged throughout the set. It's not a fluke. It's many systems. But, and that's important as well, at long times, this is a good message. Everything works. So if you are willing, and again, these are small systems, to go long, 500 nanoseconds, for example, they are all getting the same result. So that's reassuring, and that's as expected. But it requires further study, try hybrid strategies, and of course, apply a variant of this on larger systems, because, yes, again, this is a first test. But we found it very surprising and interesting. Next example, what time is it? Okay, I'm straight in the middle. Folding, protein folding. So this is another problem that is close to my heart. It's been done in collaboration, again, with Simone and Nicola here in the audience, but mostly with Margot, that was a PhD with us in Geneva, now is in the group of Max Bonhomme in Paris. And by a strike of luck, it was accepted recently, and it's been published on the journal ASP page yesterday. So you can find it if you log in and go to the latest papers. And I will start actually from the literature, actually, but not the standard literature. I was reading last winter, I was working on this, and I was reading some old classics, and I had missed this very beautiful book by Italo Calvino, Le città invisibili. And this part of an imaginary dialogue between Marco Polo and Kublai Khan struck me as very fitting to this problem, so I tried to start the article with this direct quotation, and it was accepted by the journal and Francesco as well, which is nice. And he speaks about the arch and the stone, so of course, it's a bit of a metaphor in my head, but about what is fundamental. So a bit like a chicken and egg, but how can you speak of a bridge? Is it more important the arch, the shape, or each stone that make it? If you change a stone, what happens? Is it the same? So it's fitting, and you will see why, because we are trying to build advanced CVs for folding, optimizing the feature set, one stone at a time, one feature at a time, with some ideas that include water, of course, and you will see, but a bottom-up approach, so not in a global way, touching the hydrogen bonds. Idea in protein folding, I agree. I mean, it's well known that the protein folding landscape is this rugged funnel, so it's more of a transition from disorder to order rather than state-by-state jumping. It could be even counterproducing to focus on each single state, because you don't know. You might see some states and some others are hidden, but they exist and they're metastable. The only state that you can be sure, I would say, is the folded one, the native structure. So bringing the analogy a bit forward, this is the last analogy of the presentation. I took a Rubik cube with a fake state I that probably is not even allowed in Rubik cube, but bear with me a little bit. The unfolded is completely random, and state I prime has got one face like that. The point that I want to make here in this other analogy is the following. Most CVs that we use, and we are all aware, would struggle a lot to distinguish I from F, because pretty much they are the same. There is a tiny difference. Trouble is that the ones that is most close transition state-wise to F is actually I prime, because in one move you go from I prime to F. But that would not fall in the right spot in any standard CV, meaning, of course, RMSD is bad for folding, we know, but not only contact maps, standards. It's very easy to end up in a situation like that with pretty much any CV. So the bad ordering would kill you in an unsampling. It's, again, like this review idea. You would get stuck in a bottleneck that is I, and to go to I prime that you need to go to, for example, before going to F, you wait forever. And that's not what you want. So to solve this, or to, in part, design a strategy to help yourself to solve this, we want to push I especially away from F. You want to clean F, that you know that is the proper state that is well known. So possibly mix it with I prime, even though there is an hydrogen bond, you take the standard contact between the oxygen and the hydrogen. You take also the angle because it's relevant, but that's okay. But the key part, I would say, is that you subtract explicitly the non-native contact, both with water, if there is any, very short range is the red switching function, and with other protein atoms. It doesn't matter if you visited them in unbiased simulations. You just subtract them. It's an old trick, actually, that I learned from my beginnings in the group of Michele, from Daniela Polino in urea, in horror chemistry, completely different word. But to handicraft CVs pays off in this way. To subtract, for example, why not? It's a very easy way, but it helps to distinguish. So if, in any case, I end up in a completely unknown of the many metastable states that are allowed, I have a penalty so that I can distinguish it better, that state, from in the Rubik cube analogy, if you want. Same, but even easier in the sidechain business. So we take the center of mass of the sidechains, take the contact, and subtract any unwanted contact. So you get the idea. And we sum them over. This is done on purpose, not that we are not aware of machine learning, mixing up many of the methods that many in the audience developed, linear, non-linear, whatever you want. No, we want to keep it simple, so that will be done later. But for the moment, we just sum it up with coefficient 1 as the stones in the bridge to see how powerful or not it is. We run first the good old Chignolin double mutant from the D-Show paper. We rerun the unbiased simulation because of some force field inconsistency between old Desmond and Gromax. We can speak about it later. The trajectory is available on Zenodo. So made our own reference. Today it's easy very fast to run that. And first, I turned off the non-native contacts. And these are 5 independent single OPES replicas, no one OPES yet. We run the full CV, so the only difference is that I have explicit non-native contacts subtracted much better. You see? Error bar is not tiny until the end, but much better agreement from a relatively short time. Nice. But the real example, actually, well, one OPES, of course, even better, but that's kind of expected, so good. We even bias some water coordination in a compatible way to the fingerprint, so there could be some intermingling of the two papers. But the real application, and this is the very last point, is tryptophan case that some of you, I spoke in the dinner and all, there is a lot of trouble. Tryptophan case is not easy. So we run our own 200 microsecond reference, fewer transitions. It's not that big, it's only 20 residues. It's effectively a very mini protein, but it's hard. And to see how hard it is in a bit of a posteriori way, if I project the scatter plot of the long unbiased reference that is again on Zenodo with standard CVs, you see that there are some warnings, let's say. RMSD on the left over the C alpha, it has a terrible ordering. You see, you are red, very unfolded on the bottom right. Then on the left, you become light blue, like the transition state. Alpha RMSD as well. It's deep blue, deep red for a big series of states, so as well. Even anti-beta RMSD, there is a red signal in that area. What's going on? So anyway, we run one OPES, long story short, it converges much better. We see events, of course, it's not as ideal the CV as in Chignoline. This is a hard problem even for our CV. So it's much better, we would say. Can it be improvable? Yes, in my opinion, it's still improvable. We are looking for ideas, still working on it. But we can say that we have residual of this structure, and so on. You see, then we have this wet molten globule, dry molten globule, folded to unfolded. You see, if you see some structures that Simone extracted, they are pretty similar, especially the last three, DMG, F2, and F. They are pretty much the same. Many CVs would have no way to distinguish them. They're at different free energies. And our CV, of course, it doesn't distinguish them amazingly, but it does distinguish them. There is a clear basins where you can extract and you can see. And only the very sharp top right basin of the folded is the one that you see have got a very well-formed alpha helix with both ends formed. There is a proper other 310 alpha helix in the middle, unlike F2 and so on. So that was quite a successful simulation that we are proud of, and of course we are going to keep improving and applying it to larger systems, mutants, and so on. But it's good that just in time for the presentation we could reach this. So I'm done. Conclusions, we have shown two examples about two biophysical examples where water CVs are very relevant. Many further improvements, of course, can be done, and with nice discussions and your help as well, with machine learning methods, for example. We are going to apply, of course, to larger systems. We in part already did GPCR activation, for example, bigger protein ligand binding, you name it. So there is no lack of ideas and bigger examples at end. And of course, I want to end thanking, of course, the old Parinello group, the snapshot that I was very proud to be. Memories and nice time. And of course, many thanks to Michele. Without you, all this would not be possible. And happy birthday again. Thanks.
Wondering if you had any chemical intuition about the first thing you presented on the bulk and anti-bulk CVs. Yeah. If you have any insight about why it works better in the...
The real question is, I'm a physicist, so I have no chemical intuition. That was a proud point of Michele that put me in my first project to do chemistry without any chemical prejudice, as he said. But of course, Mark is a real chemist. He can speak to you about the intuition in the poster, and on my own, I would say it's a bit of a complex situation. So some is obvious. So oxygens charge tend to be more bulk, of course. Carbons that are hidden are more anti-bulk, and that's fine, but not always. So there are some surprises if you go into the little details of all those, because you are measuring with this fingerprint the local environment in an MD simulation. So it could be that, of course, due to force field and all, you might see some unexpected behavior. So some atoms that are, by the old chemical book, hydrophobic, maybe they are more hydrophilic than expected, and the other way around. And I give a very average picture on purpose here because we want to see the average effect and the automatic strategy works. But going deeper in each system would be very rewarding and would be very nice to discuss. It's a good point. So no, you're very invited to speak with Mark in the poster, case by case. Yeah.
Ali.
Thanks, Valerio. Very interesting. So I understand that including these hydration CVs improves convergence.
Yeah. Somehow in these more systems, I mean,
but
yeah.
Can you comment a bit on is adding the hydration changing the barriers along specific directions, or is it there's a role of friction induced? So yeah, where's the physics?
Next to your atom, and hydrophobic, mostly atom, and then you jump in, and then continuously you keep moving. So you are diffusing better in the liquid by this sort of bubble increased rate. We couldn't prove it. This is complete But it would be super interesting to see, because we are really surprised. It shouldn't work better, I agree. But
I think it also depends on the system. In your case, that one is often exposed, the binding site. So in that sense, the solvation depends where is the barriers. But depends on the system. Buried binding site, you may have more.
Absolutely, no, 100%. Applied to larger systems, this really
all
shouldn't
you need
work.
is water and something also, maybe.
All you need is water, asterisk, in this more as gas for the moment, but some variation of this, given how thought-provoking, could be useful.
The key message is that don't neglect salvation, of course, the water in a binding simulation, that's sure. Thank you very much again, Valerio.
Thank you, Vittorio.
The next speaker. Don't remember the name, sorry. It's Narius Ansari, okay?
It is a fast and accurate method to calculate absolute binding free energy for ligand protein binding or any kind of other targets. The content of my talk will be first short introduction about the importance of the study, which method to use. The first part of the result about ligand RNA binding, a challenging case. I will explain about the acceleration issue and what was our solution. The second part of the result about ligand protein binding and conclusion. As you know, our body is full of tiny, very complicated biological machines. Let's call them RNA, DNA, or protein, and their countless job makes we feel happy and healthy. But as soon as even one of them start to misbehave, we will feel sick. So we need to correct this issue, and one very powerful approach to fix this issue is to use what we call a small molecule drug. Now how it's work? First, for any kind of the target, we need to precisely identify the active site, and then we need to design the ligand that can enter in the binding pocket, fit, and it's not only when it's fit, it's completely get bound to the pocket, and it means that to block the active site. So in the language of statistical mechanics, we have a target, we have a bunch of the ligands, and we are interested to calculate free energy surface of the ligand when it's in bonded and unbonded state. And the winner for us will be the ligand with highest energy gap between bonded and unbonded. But if we think about even weak binders, usually this energy gap is around few kilocalories per mole. It can take a microsecond or millisecond of plain MD simulations. So the need to use enhanced sample variable from simple distance to angle, torsion, water, or more complicated data-driven deep learning series. But imagine about the level of work in drug discovery company. We are dealing with tens of the ligand, and for each of them, there is 1,000 potential candidates that we need to calculate free energy surface to, in the end, identify only one hit or lead. So the use of more general and friendly collective variable is really essential. This lets us to use different kind of collective variable, which is the interaction of the ligand with its environment. This collective variable is very, very universal, and we don't need to introduce any specificity in this collective variable. But how does it work in practice? We can introduce a non-physical parameters called lambda in the Hamiltonian of the system, and this lambda control the strength of the interaction between the ligand and protein. Lambda is between 1 and 0. When it's 1, it means that we have fully interactive environment. And for the ligand binding case, we call this state as hollow. And when it's 0, it means that to the lambda. So lambda will have its own kinetic dynamic method. Since this method is alchemical method, so there is a thermodynamic cycle, there is an alchemical path to decide how to turn on off the thing will be wrong. So we need to have a closed thermodynamic cycle. The solution is that to add a restraint on the ligand, even when there is no interaction between the ligand and protein, to keep it in the binding pocket, to have a proper thermodynamic cycle, and in the end, calculate what is the bias that we apply for this restraint. In the literature, there are many different ways of applying the restraint, and what we used in our simulation, the restraint calls distance to bond configurations, or DBC. This is a collective variable that in general measures the RMSD of the ligand with respect to equilibrated structure of the protein. And it's important that this restraint is both transitional and orientational. So in our simulations, lambda is collective variable, and the enhanced sampling method that we first start to use is adaptive biasing force, ABF, and this method simply memorizes the force that applied on the collective variables and apply the opposite of that to flatten the free energy surface. And in the end, we can recover phase through thermodynamic integration. We start to use this method for, because RNA is a key player for many diseases, and recently there is a new therapeutic strategy that they target RNA instead of protein. But computationally, it's a little challenging because RNA is highly flexible and highly charged. To do our simulation, we used amoeba polarizable force field, which is more relevant for this system because it's charged, using TinkerHP code and Calvar's library. Complexity in this system because there are 3 magnesium ions that stabilize the structure of the RNA, so more charge in the system, and also all the ligands that were studied in the paper are positively charged, 2+ or 3+. So again, another layer of complexity. If I back to lambda dynamic again, since we will have continuous switch between the apo and hollow states, and since we need to have a closed thermodynamic cycle, it's very important to see if during the changing from apo and hollow, is there any conformational change in the structure of the protein or RNA or not. To that, we checked for this ribo switch experimentally. It's shown that RNA it has a bent structure in the apo states, but as soon as ligand gets close to that and bind to that, it's induced conformational change, and RNA gets more straight structure. But this conformational change is not only in the backbone of the RNA, also there is a rearrangement of the magnesium ions. So before running any lambda ABF simulation, it was essential to know what is the barrier associated with this conformational change. Is it doable to reach to this conformational change during lambda ABF or not? Since this conformational change is really collective motion, and it's very hard to use single CV to describe this, we used deep LDA CV that developed with Luigi and Michele to get single CV that can simulate the collective motion. To train the neural network, we used different possible distance and angles between phosphate atoms and magnesium ions. In general, we use the 47 per mole. We see this conformational change. Here you can see RMSD of the RNA, and it goes from the apo to holo state. Since 13 kilocalories per mole is huge barrier, it's impossible to access such conformational change during fast lambda ABF simulation. So to simplify, we decide to put positional restraint on the phosphate atoms and magnesium ions. So always we will be in holo state, and in the end, we can add correction in the final result. And here you can see our result, the correlation of the experiment with the simulations. Considering the difficulty of this system, we can say we have very good correlation. RMSE is one of the criteria, is around 1 kilocalorie per mole. And importantly, the method was able to identify 9 of the ligands as 9 binder in good agreement with experiment. Also, since we had the raw data of lambda ABF and experimental data, we calculate the apo-holo conformational change barrier using our data, and we find 10.3 kilocalorie per mole. That is very good, in very good agreement with what we get from deep LDA. So we were happy. Okay, we have a good pipeline to target RNA, but there is issue. ABF alone has limitation in the full exploration of free energy surface. As you can see here, imagine this is our free energy surface. ABF flatten the top part of the free energy surface, but it can easily can get stuck in the local minima, which will influence the convergence. Again, if I back to company level, we are dealing with many simulations. So we cannot run weeks or months just to get one number for free energy. So we need to use a fast, of course, OPES Explorer, because it's effectively efficient in the exploration and slowing convergence. So we mix these two methods, and we end up with lambda ABF OPES, which you will see how it's improved the result. How it works in practice, imagine this is the trajectory of lambda versus time when we apply only ABF bias. And system always was getting stuck around 0.2. When we applied even a small magnitude of bias with OPES Explorer, we had perfect exploration of the lambda space. So the question was, what is happening around lambda equal to 0.2? What is the orthogonal degree of freedom, and how OPES is helping? Okay, this is a great point, back to what Valerio mentioned, water maybe, because in lambda ABF, we have again continuous exchange between the apo and holo. So when ligand disappear, there is a water, then water should disappear, ligand come back. So we did deep analysis on the trajectories of lambda ABF OPES by looking at the coordination number of the water in the binding pocket. And surprisingly, we see that exactly around 0.2, we have a change in the number of waters when we go from holo to apo states. And it's exactly correspond to what is happening in the lambda exploration. So how OPES is working without explicitly putting any collective variable that is related to the water. It's just helping in the exploration of the water space. So it's very simple. There is no parameters to tune, and it's working very well. We applied this method at very good correlation. But this part is not what we were happy about, because using other method also there is very good correlation for these systems. What we were very happy is the convergence time. Here, if you see, this is the convergence time for complex phase and solvent phase in the electrostatic and van der Waals leg. And per Walker, we use 4 Walker in our simulations. And in average, we could reach convergence by considering all the Walkers around 100 nanoseconds, which is at the moment one of the fast methods to get absolute binding free energy. And by considering that In the real drug discovery project, the ligands are really complicated, the targets are really complicated, and this method is working very well. And here you can see the comparison of convergence time when we have lambda ABF and lambda ABF OPS. For example, in the electrostatic leg, using lambda ABF, the convergence happened around 3 nanoseconds. Lambda ABF OPS, we reached exactly the same value around 0.3 nanosecond. Or for the van der Waals leg, which is more complicated, using lambda ABF alone, we reach around 77 nanosecond, but using lambda ABF OPS, we reach the convergence around 16 nanosecond. So as a conclusion, I can say lambda ABF OPS achieves up to 9-fold improvement in the sampling efficiency and computational speed without adding any complicated descriptors or CVs in the simulations. This part is my scientific story, and if I say something about my story about how I get to know Michele, the story starts from ICTP. 9 years ago, I joined ICTP where I was looking for another position. So one day, Ali came, Ali told me, oh, Narges, Michele is coming. Maybe I will introduce you to Michele. Then Michele came for 2 days. The first day passed. I was waiting. No news from Ali. Second day, at 5 p.m., Ali texts me, Narges, come to my office. You need to chat with Michele. Oh, a lot of stress. We went to there is some red sofa in front of the coffee bar. We went there. I showed Michele some of my slides that we worked with Ali and Alexandro. And after 5 minutes, Michele, with a smiling face, he told me, did you make these slides? They are very beautiful. Then he just explained me about the Lugano, how it will be good. There are many good people. And this is how I joined to Michele's group. I had really, really good time in Lugano and Genova. And as a fun, when I was in Michele's group, always if some people were asking me to do something, I was asking €5. And in the end, they gave me all of €5. But now I noticed that I was asking €5, but they gave me, I think this paper is for me as a lucky paper. I have it in my wallet now for many years. Thank you, Michele, and also thank you, Ali and Alexandro, that you pushed me in this path. And sorry, just I would like to thank Qubit. I'm working in Qubit Pharmaceutical, the great group there with different people with different background, and it's a nice place to work. And in the end, if you are interested.
Okay, thanks. Hey, Narges, amazing talk, and as usual, the colors and the movies. I have a quick query related to the RNA ligand binding. So let's say when you truncate the RNA, let's say you take a small piece of RNA, essentially we increase the conformational flexibility compared to if you take the entire RNA sequence, let's say. I mean, it's an imagination kind of thing. So what would you comment on this? Like taking a longer RNA where the conformational flexibility will be restricted, and when you take the smaller one, then essentially you're increasing the conformational space. That eventually impacts your ligand binding.
Right. Very great question. Thank you. Yes, it's right. But the trick was to, since we cut the RNA, the trick in the literature even is to put some position in distance restraint in the tails of the RNA to not let the RNA to open. Otherwise, it will start to open during the simulations.
Okay.
And since in the experimental paper, they tried two sides of the RNA, 29 member and 40 member.
Okay.
And there was no difference in the final result. So we had confidence that this size is acceptable.
So eventually you include this thing during reweighting the free energy surface, right?
Exactly.
And what is the typical, let's say, simulation time scale within which it converges?
Yeah, for the lambda ABF opus, it's around lambda ABF opus 100 nanosecond with 4 Walker.
Okay.
And if you are asking about the performance, since I'm using amoeba polarizable force field, which is 10 times more expensive than the point charge, so it depends on the GPU. Nowadays, with the fast GPUs, it's super fast. And the ABF opus, but plain ND is doubly.
So 16.
16, exactly.
Okay.
But using H100, it was 36, 40 nanoseconds.
Okay. And the second question is, how are you getting the bound structure for the RNA and the ligand?
For the RNA, we had the X-ray structure for one of the ligands. So we start from that X-ray structure. And since the binding pose of all the ligands was the same, we used that one to reproduce the binding pose for the rest.
Okay, and follow-up question. Going ahead for new ligands and new RNAs, how do you envision doing that when you don't have any extra structure? How will you start the simulation?
The big, I can say, bottleneck of this method that we need to know the binding pose, otherwise it is impossible to use it.
Pratyush just made the question that I have in mind. And for when you mention about heat screening, heat discovery part, so you have any plans how to do that in your company when you don't know the binding mode, as Pratyush was saying? So are you going to switch to another methodology, I guess? Right?
We are using at the moment this method, but you know that there are many steps before final ABFE. There is virtual screening, docking, there is some potential candidate that we use this method in the end.
Okay, thank you. Marco.
Sorry, I made you. Okay. Okay, thank you. It was very interesting. And we also work a bit on, more than a bit, on RNA and small molecules. And these are very challenging systems, as the first question pointed out. So I was going back to that point and see if you can share with us when you have been useful, for example, to restrain the system, or do you leave the system completely free to sample states? How do you manage that aspect?
Thank you for the question. Yes, initially we didn't put any restraint, but it's super challenging. Choose each of them, calculate the binding affinity for each, and in the end have an average overall. But we were lucky in this system because we had X-ray structure, so it was very easy to know what is the stable structure.
Thank you.
There are other questions? Not the case. Let us thank again very much for the presentation.
Non funzioni ancora.
L'appuntatore per quello piccolo.
Ok. Ok. Good.
Okay, it's a great pleasure for me to introduce you the last speaker of this morning session. He's a colleague, friend, Alessandro Barducci, and from Montpellier. And the title of his talk is Modeling Biomolecular Structure Interactions in Cellular Condensates. Please.
Okay, so good morning, everybody. I'm really happy to be here. And, I know that I'm basically what stands between you and lunch, but apart from thanking the organizer, I really want to start by a child, a different age, but we didn't hear anything about you as a student, if I'm not wrong. So, everybody knows you're a brilliant researcher, so I think it's easy to guess that you must have been a brilliant, possibly exceptional student. I cannot add much on that, but I can add one small detail. I know that luckily, at least for me, you were not a perfect student. So why I know that? I joined Michele Group in 2007, and I applied to the Enrico Fermi Center in Rome for a fellowship. And, you know, the application procedure, the selection procedure was not, how can I say, crystal clear. And so after months of waiting with all sorts of short-term contacts and so on, Michele eventually had to pick up the phone and call the director and founder of Enrico Fermi Center, who was none than Antonino Zichichi, who for the non-Italian is another very famous Sicilian physicist. And it turned out that Michele actually had taken a class with Antonino Zichichi back in Bologna in the 60s. Class was Istituzioni Fisica Teorica, if I'm not sure if I remember correctly. And of course, he got full marks, but not the cum laude. So during the phone call, he reminded Zichichi of this detail, and Zichichi must have regretted it a little bit. So, of course, the grade could not be changed, but the fellowship could be awarded. In a sense, I owe my career to this small imperfection of Michele as a student. I'm very thankful for that. Okay. Now that I shared with you this small story, let me talk a little bit about science. So in the last year, I'm working in a center of biophysics and structure that this compartmentalization does not rely just on the textbook organelles like the nucleus or mitochondria or so on, but also on this kind of dynamic assemblies of proteins and nucleic acids. And these guys are basically ubiquitous, so you can find them in all compartments of the cell. Some of them are constitutive, some of them are formed and is assembled depending on the condition, for example, with stress and so on. There is a lot of speculation about how they actually work, but we know that they are fundamental, and we are finding more and more and more. I mean, most of the interest, at least for physicists and chemists, for these membraneless organelles was driven by the fact that, at least for some of them, I mean, their assembly could be interpreted with a simple physical model, like liquid-liquid phase separation. And this attracted a legion of soft matter physicists to the field, and this simple concept has taken cell biology by storm. So that now, I mean, people like interdisciplinary research, there is a lot of interest on this topic. Of course, this analogy of oil and water showed also some limitations, because these assemblies are actually made up mostly of disordered proteins and flexible molecules like RNAs, and you have a lot of different interaction, basically a dynamical network of intermolecular interaction, relying on both simple electrostatics, but also folded interaction and so on. So it's a complicated story. It's actually not liquid in a physical sense. From a physical point of view, they show more some kind of gel state, and then they are intrinsically coupled to the formation of solid assemblies like amyloids, and most of the people working on aggregation and protein aggregation are actually now considering these liquid condensates as some kind of metastable preliminary step before going to aggregation if the process is misregulated. And it's a bit organized. Then there is like this interplay with irreversible aggregation. And then something that has been somehow not fully appreciated to me in the field is that in the cell, you have a lot of non-equilibrium processes. So eventually, equilibrium statistical mechanics is possibly not always the best tool to understand how these objects are assembled and regulated. And I mean, we are trying to use many different molecular simulation approaches to investigate these objects and trying to complement experiments and try to address some of the questions. And of course, they're big objects, so we rely on good old atomistic simulation, but also coarse-grained approach that can be used to study real phase coexistence, so to trace phase diagrams of these, for example, disordered proteins, but also phenomenological coarse-grained models, sometimes coupled with Monte Carlo in a way that is reminiscent to the ultra-coarse-grained method, developed by Greg Voth. And I mean, I'll try to stick to the title of this conference, so I will focus on good old atomistic simulation, even if as a disclaimer, I will tell you that there is not much methodological development in my group going on in this case. So as you, I mean, dealing with like collective interactions of large size flexible biomolecules, you can easily guess that atomistic simulation, solid atom simulation, are probably the easiest tool to use. In the literature, you find some examples of these studies where they try to study phase coexistence. Usually, they try to study phase coexistence. Usually, the recipe is like that. You use coarse grain, you build some kind of coarse grain model of condensates, and you back map at the atomistic level, and then you study how water and ions move and how proteins move. But I mean, these are large proteins, and usually the time scale is much beyond, the excision time scale is much beyond the microsecond time scale. That is what you usually can achieve even with large computers. Between different fragments of the protein, both in terms of homotypic interactions or mixing them in terms of heterotypic, and trying to see, I mean, if at least atomistic simulation could contribute to discovering this kind of molecular grammar, which residues are more important, and so on. And so, of course, with atomistic simulation, you can easily try to see if you get this kind, for example, arginine aromatic acid interaction considered to be very important on the basis of mutagenesis studies. We could confirm that simulation could reproduce these results and also play some kind of computational mutagenesis tricks. And of course, with simulation, you can really see, I mean, atomistic simulation, you can really see what's going on. So we could identify these stacking interactions and then T-shaped interactions, and also identify that you might have higher order oligomers of aromatic and planar side chains that have not yet been discovered, but we're discussing with NMRs to see if they're actually observed. Okay, that was a simple strategy. So fragments in order to avoid the timescale problems and having to have tractable simulation. That's something we can extend. We extended with Giovanni also to the interaction between small peptides and short oligonucleotides to, for example, rationalize why polyarginine could form in test tubes condensates that are much more viscous than polylysine. And of course, we could explain the multivalency of arginine play a role in these different physical properties at the mesoscale. And more recently, we started collaborating with Evan Sproit in the Netherlands, who is a brilliant physical chemist who managed to find an NMR assay to study how ions are binding to proteins and modify the properties of in vitro condensates. These are basically conservates made up of a long positively charged peptide with ATP. And with NMR, you can identify the shift in the peaks corresponding to some protons, or for example, this peptide, that ion binding, since these are like large and flexible moieties, depends on the structure, and you have cooperative effects, including both side chain and backbone, and so on. Now we are trying to understand a little bit better how the ions can actually play a different role in tuning the physicochemical properties of the droplets. Again, these are very simple applications of MD to a field that is kind of difficult to be treated with atomistic MD. But maybe the only story, most interesting story I can tell you at atomistic level, something about the biomolecular structure. So these guys, at least in the test tube, you can have phase separation with high densities with sim-model proteins. So you might have a protein density maybe 40% of a droplets in terms of mass is made up of a protein. So in terms of physical chemistry, you can expect that you have a very different environment. And one might wonder if this environment can actually affect the structure of biomolecules. So are they folded? Do they change structure? This is important, especially if we consider molecules with a very complicated landscape, like RNAs, for example. And this landscape and this conformational variability, I mean, is deeply connected with the function, with the biological function. So condensates, in a way, might be a way, for example, for operating some kind of conformational selection, favoring some conformation, and then dictating some functional properties. So in order, I mean, we have some experimental evidence, again, very indirect, with model some nucleic acid duplexes. They seem to melt or to be less stable when they are in condensed phase. like have some kind of unfolding driven by the environment of the condensate. Again, talking short RNA hairpin that are guinea pigs for people interested in the structure, and they are small, so like 10 bases, but they have a well-defined folded structure solved by NMR in solution. So using our fragment-based idea, so okay, maybe we can mimic the physicochemical environment of a condensate, putting these peptides at a concentration that is reminiscent of what you had in a in vitro condensate, and using a small peptide so that you have less problem in terms of sampling as a solvent that shares a motif that is commonly observed in real cellular condensates. So, well, of course, you need some help from enhanced sampling methods. You have used a robust protocol that maybe now it's a little bit old school, but still it works beautifully for RNAs. So we combined well-tempered metadynamics with solid tempering. And using this approach has been already used by many people, I think including Michele as well, with Kressel and Larsen. We could solve the free energy landscape. So this is our test. Our hairpin free energy landscape as a function of solvent accessible surface, so how much it's open, and this ERMSD metric proposed by Giovanni that basically measures the distance from a reference structure that in this case is the NMR native structure. And you can see that, I mean, in water you have this nice structure, and you can try to extract the delta G of folding from this simulation, and we have an agreement with experiments that, using a word that I learned from Michele, it's an embarrassing agreement with experiments. In the sense that probably it's due to a cancellation of errors, but I mean, we are very happy that. So, satisfied by this kind of protocol, we extended it to our, let's say, mixed solvents, where now you can try to see, for example, which are the groups of RNA that are more responsible for this expansion. Showing here the SASA just of these groups. You can see that upon expansion, the exposure of sugar and phosphate doesn't change much. What's really relevant are bases. So the nucleobases are actually better solvated and more open when they are in this model condensate. And then, of course, you can look at tiny structural details. So this guy is made up of a stem part with 3 base pair and a loop that is more dynamic, even in water. You can try to see, for example, what's the free energy of formation for this base pair, and they are all very stable in water, but they are marginally stable or unstable in our model solvent. So you can try to make another coordinate just to show that, you know, the completely the secondary structure in the stem is stable in water, but not in our model condensates. And then you can look at the loop, and here again, thanks to the help of Giovanni, who knows all these classifications of RNA structures, we could identify patterns of staking, and base pairing that are summarized in this pictogram here. And in water, we see a kind of flexibility that is compatible to what is expected for this system. When we move to our model condensates, basically what we see is that most of these interactions are destabilized, but surprisingly, you also have the formation, partial formation and non-native pairing, suggesting that it's not just destabilization, and possibly in much larger and more complex RNA structures, a different chemical environment could actually stabilize alternative conformation, paving the way for, you know, possible new function. And then, of course, I mean, usually at this point, the biologists in the audience ask me, so have you done it for how many condensates? Actually, something intermediate between what we get with the first peptide and what you have in water. So the destabilization is less pronounced. And this is, again, in good agreement with, qualitative agreement with experimental data. Now we are trying to get our dedicated experiments just to show that agreement is more than just qualitative. And in terms of explanation, but it's not so surprise that arginine is actually a better solvent than lysine-based peptides, because arginine groups, guanidinium, they can form more hydrogen bonds than lysine, and then they have this possibility of forming stack interaction with nucleobases as well. So providing an additional mechanism for stabilizing the open conformations and eventually destabilizing the native fold. Okay, I don't know what time is it, but I know that it's time for lunch soon. So these are basically, today I talked just about atomistic simulation, but I hope that I could convey the message that using some kind of smart tricks or enhanced sampling, we can still learn a lot about complex biological, biomolecular systems such as condensates. With that, of course, I want to thank the group, particularly the guys who did the work, collaborators for the work that I showed you, as I said, Giovanni Bussi and Evan Sprite, all the funding bodies, we are hiring as well, and thank you for your attention, and happy birthday again, Michele.
Thank you. Thank you, Alessandro. Very nice, interesting results. Question from Michele.
Wow.
About Zikiki, I guess.
About Zikiki, yeah.
No, Zikiki, I had enough of him. Okay, so no, I found very, very interesting the paper because in my view is a step in the correct direction. So you were mentioning that there is a phase equilibrium. Is that for the system you're studying? Have you tried to move to the critical point where mixing the mixing thing?
We have tried at the coarse grain level, we have tried, and there are several, I mean, it has not been explored in a stat mech point of view, because usually these points are, I mean, experimentally you are kind of far from the critical point.
I don't mean that you want to study the critical property, the indices. I don't think it's. It would be interesting.
No, you can do it,
but putting yourself close to that region, where
where you can have large scale fluctuations, and we have tried. But as you know, it's the point where simulations get a little bit more complicated also in terms of sides and so on. But it's something that you are exploring, but they have not results for the moment.
You don't need, no, that's what I say. Don't need studying the critical point. I understand there is a critical slowing down and so on and so forth, but there is a preclinical region where all these things don't happen, and you can look at it. Or
the real point is that, I mean, at the all atom level, clearly this is still not possible because you don't get a real demixing with peptides. The point is, what is a good control parameter in terms of coarse grain simulation? Because you know, coarse grain simulation, you change the temperature, you're not actually changing the temperature. You're changing the coarse grain function in a way. But that's something, you know, still unexplored. But
so thanks, Alessandro. I think this is a really exciting and interesting topic.
Wow.
Yeah. The experiment.
In vitro, you take a couple of components, you have a perfect liquid phase equation with your nice round droplets, you measure surface tension, whatever. When you're in the cell, everything becomes more complicated. And there is like a huge gap in between where we are missing some in vitro controllable model that still has some degrees of complexity that is reminiscent of the cell. So particularly for non-equilibrium system, you can add kinases, phosphatase enzyme doing works and so on. In terms of techniques, of course, it depends if you work in vitro or in vivo, but microscopy and fluorescence are very popular method, particularly because you can address things in the cell directly. People have tried to trace phase diagrams in the cell, but of course it's difficult to control concentrations and so on. But yeah.
I see. Interesting.
Alessandro, a follow-up question to these two that I heard. The two very short questions, actually. First one, the physiological meaning of that peptide. Why is known to be related to some mimicking, modeling of what happens in vitro, I guess, but probably I lost this message.
It's a very good point. So, I mean, it's a brute crude approximation of, I mean, you have many RNA binding proteins that have disordered parts. That particular sequence is enriched in these like disordered regions, low complexity regions. That's why we chose this one. And then, of course, this is something you can do in real experiments.
Okay, so it's
In in vitro
model
experiment.
that...
It's an in vitro computational model for
and related to this, you showed how the results change, very interesting, different states, also the graph that you use also with the ad of Giovanni, in the using different peptides in which you just mutate arginine to lysine, okay? Naturally, people who works with nucleic acids, it's net charge plays a rules. So did you use classical description of the net charge between arginines and phosphates, or you found that you...
Different pH inside condensates. You might have many things going on with electrostatics. It turns out that eventually we are still stuck to good old, because I mean, there are, no, maybe amoeba could be like, I mean, somebody should start using, because I mean, those are very good questions. We don't know.
It's a point known. Just I wasn't if you just took this point or somehow.
No. We'd like to test some polarizable version because I think they might.
And in any case, the comparison between arginine and lysine, since you are using the same approximation, you are just comparing.
I'm kind of confident that this would not change much. But
relative differences, yeah. There's another question for Alessandro. Otherwise, there's one. Yeah.
No lunch.
Hi, Viridiana from Mexico. Thank you for your presentation. I have one question. I see that this might have applications to the study of neuroscience, for example. So my question is regarding that. Many neurodegenerative diseases are associated with this protein aggregation. So this concentration of peptides in cells, is it known if these type of peptides are present in these specific diseases, or it's just a model?
This particular peptide, no, I will not claim that it's connected to a specific neurodegenerative disease. The point is that this kind of liquid state of aggregation is intrinsically, connected to aggregation, because it turns out that most of these condensates, even the model in vitro one in a test tube, will age, will undergo a process where, slowly or fast, they will turn into some amyloid state that can also self-propagate. So this is like a big, from a physical point of view, a big challenge, I think, in the field.
Okay, I don't see any other hand raised. So let me thank Alessandro and all the speakers of this morning session again with a clap, with applause. And now it's time.
Beto. Grazie.
beautiful picture that give a static image. Here is a SARS-CoV-2 virus in the cell, but we know very well, people in this audience, that dynamic biology is messy, everything moves, and really to understand how complex biological system move. And this is the spike of SARS-CoV-2. I'm sure you are very familiar with it. And we are especially interested to a class of systems that are extremely dynamic to the limit of not having a proper structure, one single structure that we are used to with globular and stable proteins. And these are called intrinsically disordered proteins or regions that are ubiquitous in biology, and they carry and perform so many functions from nuclear transport to be with Michele, while first converge, almost converge, free energy, converge for 2006, free energy landscape of a small peptide. And we all know what are the limits and the challenges in using this kind of tool, the time scale that you have to simulate, the accuracy of a force field. And we are trying to address these problems in a kind of an orthogonal way in what we have been used to with Michele. And the idea is to not just use a theoretical description of our system, a force field, but to integrate during the generation of our biological dynamics other types of information, especially experimental data. In particular, structural biology experiment like NMR and so on and so forth. The idea is that we would like to address the usual concern of the reviewer. Did you try running the simulation with another force field? And our ideal answer is yes, but the result that we obtain are force field independent. So no matter where we start from the point of view of the theory, we can inject other information in our computer so that what we obtain at the end of the day do not really depend too much on the physical chemical details of our force field. So this is where we aim, what we aim to do. And if I have really to pick a point in time when I developed this desire to relate an in silico world to the real world of experiment is exactly in the lab of Michele. It was almost 20 years ago. These are kind of serious pictures. I joined, I think, in beginning 2006, and this is one of the first. It was not there, so I had to add it with Photoshop this morning, but it was always there in spirit. And this is the composition of the group at that time. So you can see we are very kind of seriously dressed, but you have to understand, me and Michele, we come from a very different origin. I'm more from the north of Italy, I would say Bergamo, and compared metadynamic simulation to experimental data. At the time, we were getting pretty good in accelerating sampling, but not so good on comparing our bias enhanced simulation with reality of experimental data. And so we had to find a way to go through this converge a free energy landscape in which you accelerate a few degrees of freedom with metadynamics to compare with other degrees of freedom, which are the experimental measurement. And in order to do so, we developed with Michele probably the first metadynamics approach to reweight a bias metadynamic simulation in order to calculate experimental observable. Here, the example is nuclear magnetic resonance. We compare to scalar coupling, and you see no matter where we start our metadynamic simulation, from a completely extended state of a folded helix, we can fit and explain very well the experimental measurement once we remove the effect of metadynamics. Instead, if you do standard MD, of course, you get stuck in local minima, and this, you know why. So this is where I started, really my desire to connect with real life. And over the years, the things become a bit more integrative, in the sense that we don't just want to compare, we want to integrate the data during the simulation. And we based all our work on some probabilistic theory in which your kind of prior expectation of the properties of a system that comes from MD or another method is updated once you obtain new information, new experimental data. So in simply speaking, instead of sampling Boltzmann distribution given your force field, you have a more sophisticated distribution in which you integrate the experimental data. You model all the errors at play, because experiment like simulation are not perfect, and we want to weight all of them based on this accuracy. At the end of the day, we have a pseudo energy function here that is just a modification of your standard force field with some additional terms that enforce agreement with the data of a whole. This is what we have been doing, and pretty happily until a few years ago, once there was this kind of revolution for us working in structural biology with methods like AlphaFold and other prediction method that simply starting from a sequence, one-dimensional sequence of amino acid, they can predict pretty accurately a single structure, a global minima of a system. And this for us changed things a lot because we said, okay, now we have also this type of information along with cryo-electron microscopy data, NMR data. How should we best use this information along with experimental information? And so how do we fit this in our virtuous cycle? And our ultimate goal really is to be able to do biology where it matters in the cell. So make use of maybe low-resolution data from tomography data, for cross-linking, with a prediction coming from artificial intelligence to be able, like Greg showed this morning, to build these complex models of gigantic protein complexes in the cell. And this for us is a quantum leap, if I think 20 years ago, where we start from 22 atoms to get to these huge systems. It will really be a challenge. So how do we do that? So everything that we do is modeling the information that you have in terms of uncertainty, probability distribution. How can we describe this new data that comes from artificial intelligence in our probabilistic framework? And the way to do it for us is start to modeling and understanding the structural in this prediction coming, for example, from AlphaFold. If you are familiar with this method, maybe you have seen these very colored pictures where the color reflects the confidence in a particular region of a protein. This is a disordered protein, FUS. And you see the orange part is completely disordered, so the prediction makes absolutely no sense. And then there is a folded core in blue in the create in the process of getting to this model. They really do predict the confidence at the level of each pairs of residues, at the distance of each pairs of residues. So this is what we call histograms. This is comes out, is produced by AlphaFold, and for each pairs of residue is a distribution of uncertainty in C-beta distance. So if you look at the single model that comes out from AlphaFold, for this specific distance is more or less there at the peak of the distribution. But sometimes the real structure that experimentalists have solved doesn't match the prediction from AlphaFold, but it falls within the uncertainty of the model. So if we are able to sample along this uncertainty distribution, we might be able to improve our models or to integrate with other data coming from experiment. And if you wonder what is the relation between these representation of uncertainties as something you're more familiar with, you can see that the broader is this distribution, the larger is the uncertainty in that specific part of the model. So this is what we've been doing in the last couple of years, thanks to nice funding from Europe. And so we were able to create a unified framework in which experimental data and prediction from artificial intelligence are treated in the very same way, without any subjective choice of what is right and what is wrong. It's just we put all the data in our computer, we let it run for days or minutes, and at the end of the day, so this is not the story of today. Because one of the things I learned with Michele is once you have a method that seems to work, well, you have to use it for anything. And try it and play and push the limit of your method until you really break your new toy. So this is what we have been trying to do. And we have been asking a weird question, what in this prediction that come from AlphaFold, there's something more than uncertainty, and AlphaFold somehow has learned something about the thermodynamic of the system or the dynamic of the system. What is this disordered system using a tool that has not been designed for that, like AlphaFold? So this was our weird question at the beginning of this project, and to address that, we use our probabilistic framework in which we integrate basic physical chemical models with the information that comes from this AlphaFold distribution. And I will go into the detail what we call bias. So there is a prior model that I will describe very quickly, and then the information coming from AlphaFold predictions. So we start really with basic physical chemistry force field. We took a pretty fantastic force field, and we remove all the nice thing of this force field. We remove electrostatic interaction, we remove water, we remove everything, and we kept an atomistic description of the system. So we have to play a little bit with this force field in order to have a kind of an unbiased coil model for these IDPs. So this is our prior baseline force field, and we want to understand if AlphaFold, this distribution, are bringing some information about IDP, when it shouldn't work at all. So we take this distribution and we kind of model a little bit to simplify, like Gaussian distribution. And since we use this formalism with experimental data, where we have a model of the error, we assume that the measurement could be wrong. We are doing exactly the same thing here. We said we have this prediction, but this prediction could be wrong. So we don't enforce exactly this distribution. We said we start from this distribution, but admit that this could be completely wrong, and we just let our alanine dipeptide, or more kind of challenging system, we want to push it a little bit more. So we selected, I don't know if you can say, around 2021 system. These are all these better than other methods. And we have also more low-resolution data, like small angles X-ray scattering. If you're not familiar with it, it gives information about the radius of gyration, about the shape of the proteins. So the objective is to be able, with our approach, to reproduce the behavior of these complex IDPs in solution with information coming from AlphaFold. So there are different classes of IDPs. The first test is with IDPs that are really, they have not a lot of structure. They really behave like random coil. And this is one example, a beta 40 is important for neurodegenerative disease. And we can compare, we can generate our structural ensemble and compare, for example, with chemical shift there, with RDCs, and with scalar coupling. So you have in gray the experimental measurement, in blue we have our prior, our coil model, and in orange our approach. So we don't want to destroy a perfect model, the coil model, for systems that are completely disordered. We want to be able to reproduce the same behavior once we introduce AlphaFold information. And this is the case when the IDPs are completely disordered. You can check across a wide range of IDPs, and the quality on average and fit with this data is exactly the same as our prior coil model. So this is a sanity check, but when there is no structure, there is not a lot there. If we introduce information from AlphaFold, we don't alter the quality of our prior. The most interesting part is when there are residual secondary structure, transient helices, we want to be able to characterize that. And so we compare quantitatively our ensemble with chemical shift data, RDC data, and SAX data. As you can see here, for example, the first plot over there, if you use a coil model with no information about secondary structure, you cannot fit absolutely and explain this solution data. But if you integrate the information from AlphaFold in the way we are integrating it, we can reproduce pretty well the behavior of this system in solution, which is pretty surprising to us. The system is a folded proteins. And these come, for example, for systems that folds in different conditions when deposited on the lipids, in presence of calcium. You get folded state when it should be folded in solution. But since we are assuming that this information could be wrong in our approach, we are able, even if the static prediction, the PDB that AlphaFold gives you is ordered, our conformational ensemble display a limited of order, but also a disorder. And you can see we can fit very well solution data in these cases. And we can check that this is the case, that is because we are modeling the errors in the data that we can fit this, explain these problematic cases. If we kind of just take the information from AlphaFold, we believe 100% in their prediction. At the end of the day, what we obtain is exactly a very folded ensemble. In this case, is alpha-synuclein. It's, again, another important protein for neurodegenerative diseases. And the data we fit is the disordered system in presence of a lipid membrane, so when it's folded. So the reason why we can overcome this limitation is because we assume that the data the prediction could be wrong. And this is true for not all, but the majority of cases we are very in orange compared to dark red. So we would like to push it a little bit further, because now we have compared what this predictor is bringing to the study of disorder system. We want to compare with state-of-the-art methods, state-of-the-art atomistic simulation, explicit solving, things that are very expensive. And we focus on 4 systems that have been extensively characterized by DSO research. So there's a big computer that can afford microsecond and microseconds of simulations. And for this, we have, again, an extensive list of experimental data that we would like to check if you can explain. So we focus on this part, and here is how it works. We have a global index of our laptop in a couple of hours instead of days and days on a specialized supercomputer. And so this is the pure simulation. As you can see these days, you can take this ensemble, we can refine using the experimental data. There are many ways to kind of adjust in a maximum entropy spirit this ensemble. And if you do that, so now we have a refined ensemble in dark color, and even after addition of experimental data for refinement, we still are competitive with state-of-the-art molecular dynamics phosphate. So this is what we have been pushing our integrative approach to do, and to our surprise is bringing some information on systems that have never been seen once the S algorithm was trained. And this should make us think what really these new methods are learning. If they're just mimicking what has been observed in the training data set, or are learning something more about the interatomic potential that might be applicable to think outside of the training set. So this is something that we need to investigate more. How much time do you have? 5 minutes. So if you don't believe me, as I didn't believe at the first time, you can reproduce everything, of course. We have been working hard to implement our technology in Plume. This is really software born during the PhD with Michele, endless nights fueled by kebab and in Lugano to create the first version with Giovanni, with Carlo Camilloni, with Davide Branduardi, and then with Garrett Ribello. And now the community expanded a lot. We have contributors that help us building other resources for Plume. I think Garrett will give an overview in the next few days. We have a repository for the protocols, and we have now a set of tutorials that you can use. And we have kind of engaged with the community a lot during the last 10, 15 years, especially thanks to SECAM funding. And here you can see a few images of our events. This is where we learned that you're covering pretty well different areas of the world. But probably the most interesting thing that we have collected over the years, thanks to your work and the things that have been created with metadynamics, is a set of amazing pictures. So we have decided to create a sort of collection of art made of Plumed. We can call it Museum of Modern Art, because you never look maybe that free energy sometimes are really surprising. And we have free energy that looks like IKEA table. This is very nice. We have burgers. These are all real free energies. We are going to the animal worlds. We have ducks made with metadynamics, with Super Mario free energy. And this is one of my favorites, which is the AT free energy. So if you have more examples, please send. We will collect and make a nice book. So I would like to thank all the people who have been working on this project and others in our group, and thanks to you for your attention.
Thank you, Max, for the very nice talk. So we have plenty of time for questions.
Great talk. I was curious regarding, I mean, you use for the IDPs as a random model something that is really random call. So it takes into account like local structure, but no long-range interactions. Yeah. So I was wondering if the information you take from AlphaFold also accounts for the deviation from this random behavior. I mean, Calvados force field that you analyzed, you have this kind of weak interaction like hydrophobic or electrostatic that will cause the protein to not to collapse, but to be more or less expanded in some regions. So I was wondering if comparing with Sachs data in the Guinea region, if you can get this kind of partial collapse from AlphaFold there.
We range, and they are both. And to get the best out of this ensemble, you need to put both information.
Well, okay.
It was surprising to me. I was thinking exactly the same. It's just little helix.
Hi, Max. Very nice talk. So I had a question about your bioarchive that you talked about today. So for example, for the disordered protein, you said AlphaFold can often make it look more folded, but then you can also do tricks like MSA subsampling, and you can force it to generate an ensemble of structures. So my question is, two questions, but there's really one. When you're generating structures from AlphaFold, it's just hypothesis, right? So you could generate an ensemble of structures by playing these tricks, and would your method still work on an ensemble of predictions? And secondly, ensemble of structural predictions from AlphaFold instead of just one structure. And secondly, could this be done on top of other AI methods like BioEmu? Could you generate structures from BioEmu and then try to reweight it and correct it?
Well, we tried BioEmu, it was one of our comparison, and it clearly doesn't work as well as other methods.
So in that comparison, did you directly compare with BioEmu output, or were you trying to reweight after BioEmu output?
No, we compared the BioEmu output.
So could you reweight after, could you correct the BioEmu output?
I'm not sure. Related to the first question, it's more complicated than that. So there are, in many of the predictions that we do, this probability distribution are multimodal. And so the question is this second pick the thing that you kind of highlight when you start playing with sequences or not? So the information is always there if you use the full MSA. It's just a minor state. It's only when you play with the sequence alignment that you can highlight that state or that state. So this is extremely important, especially for kind of discrete conformational changes. And we are investigating into that. Yeah.
Thank you. I think that when talking about intrinsically disordered proteins, your methodology has proven to be very efficient. But have you tried, for example, on very well-known proteins, for example, tubulins with a very conserved structure, but with a mutation? Because we have this problem when we try with AlphaFold to create a new structure with just a single mutation, we basically obtain the same protein that the crystal.
We haven't tried that. We are interested. What we are looking at is more kind of more native small fluctuation of globular proteins around the native state. We are looking at phosphorylation, the effect of phosphorylation on IDPs, because we can do this with AlphaFold3, so modeling phosphorylation explicitly, but we haven't tried mutation. But it might very well be that the output that you look at, which is a PDB, a structure, is identical, but the actual prediction that comes from this probability distribution is slightly different. And if you sample more extensively this distribution, you start seeing differences. I cannot answer to that question. We haven't tried, but it could be possible.
Okay. We are looking for alternatives. We are trying some other software or even trying to develop some other software because we cannot solve that question with AlphaFold, for example.
There are no other questions. I had a question.
Go for it.
So it's very interesting what you were showing, that this IDP depend on really the environment. So
landscape, a little bit of residual secondary structure. How you adapt to different situations, this is probably where the best approach is to refine this ensemble that are good with specific experimental data in your conditions. This is the easiest way to do it, probably.
Thank you. So if there are no further questions, we thank Max again. And
Not a great start. Okay. Better? Yes. Okay. Well, thank you very much for letting me crash your conference. It's very nice to be here, although somewhat surreal, I have to say, because Last time I was here was probably about 40 years ago, and I was a little kid running around among scientists. A full circle kind of moment, so thank you for that. And apologies, I will not be talking about any molecular dynamics today. I'm a biologist, and I will tell you a little bit about what we do. But I hope that by the end of it, you'll be somewhat interested, and also maybe there'll be scope for some, who knows, collaboration, or I hope it's an example of real-life problems that maybe some of your methods could be suitable to. So my lab works specifically on a type of brain tumor that is known as glioblastoma. And this is just a bit of introduction about this. This is a really terrible and that we give. So the therapies that we give to these patients, which are universal, they're the same no matter which patients they're given to, are largely ineffective. And these are a combination of surgery followed by chemotherapy and radiation. So they're really aggressive treatments, but they don't work very well. And so I think this is clear from this. This does not work. Does it work? No? Pointer? No. Okay. Maybe I can do it from here. Yes. So in this longitudinal scans from a patient, you can see that the therapy initially has some effect, but very quickly the disease recurs. Yes. Okay. All right. So, and in addition to that, I think another problem with this type of tumor is that because it affects the brain, patients also experience highly debilitating symptoms, cognitive problems, and physical sort of movement problems. So it's really a nasty disease, and we need to do a lot better to treat it more effectively. And so one of the major problems in this disease is that we typically diagnose it and treat it at a very late stage. So if you look at these cartoon here, which is representing sort of the evolution of typical cancer. It starts with cells of origin accumulating mutations, then there is a slow phase in which the tumor progresses, and then it reaches a sort of advanced stage in which is very large and very complex. So this is shown here by these different lines in different colors. So by the time the tumor is found, it's highly complicated and heterogeneous, and it develops a series of mechanisms that make it highly resistant to these treatments that we give patients. And so because of this complexity, it's very difficult to cure this disease. And you could argue that by the time we intervene at this stage, it's probably a little bit too late. And so the question really is, can we go a little bit earlier during these earlier phases in which the tumor is not quite as complex and likely to respond to treatment a lot better? And can we detect these tumors earlier and treat them earlier? The work that has gone on in my lab over the last few years has really tried to tackle this question. Can we understand early disease biology, and will this give us opportunities for more effective intervention? And before I go on to tell you a little bit about our work, I think it's important to understand the disease, that I give you a bit of an introduction about neural stem cells and how the brain develops, because you will see this is really linked to glioblastoma. So during development, the brain forms from cells known as neural stem cells, which are a bit more primitive, and they have the ability to give rise to many different types of mature cells, particularly neurons, which I'm sure you're all aware what neurons are, but these are the cells that are electrically excitable and connect to each other through long projections known as axons, and then synapses, and in this way they form neural circuits that give our brain the ability to function. And in addition to that, they also form two types of additional cells, which are called astrocytes and oligodendrocytes. Astrocytes are very special cells that allow and help sort of the electrical connectivity between neurons, but also have a variety of different functions. And the oligodendrocytes are cells that produce myelin, which is an insulation layer of lipids that wrap around the axons and allow the electrical conduction of the nerve impulses, a bit like an electrical cable, which is surrounded by plastic. So in the same kind of way. So this happens when the brain is formed, and for many years, decades actually, number of neurons and other cell types, and if we lose them, that's it, there will be no generation of new cells. And this largely comes from work of the very famous Ramon Cajal, who has drawn all these beautiful pictures of the brain from anatomical sections, and that has really given the impression that the brain is really static and the cells that are there are fixed in time. But around the 1960s, this dogma was challenged, and we now know that this is not the case, but actually in the adult brain there are some neural stem cells. And so after that, it was discovered that neural stem cells are also found in adult brains of most species, and including mammals and humans. And the neural stem cells are found in specific parts of the brain. Particularly relevant to glioblastoma is the subventricular zone, which is shown here. The details are not very important, but in the mouse, what happens is that the neural stem cells are typically just forming new neurons continuously, and these neurons go to a region of the brain known as the olfactory bulb, which is the region that allows the mouse to smell and learn new smells. But if the brain is injured, these neural stem cells can come into action and actually begin to move around and migrate to the site of injury, where they attempt to repair the brain. Now, this is not very effective, but they are trying to do that, and they can form new neurons as well as new astrocytes and oligodendrocytes to repair the myelin that is lost, and so on. So there is a lot more plasticity and potential for regeneration in the brain than we anticipated. And I think with that comes a degree of vulnerability, and you will see how this connects to the glioblastoma in a minute. In addition to that, another cell type that is quite plastic in the adult brain is the astrocytes, which I described to you just earlier, this green cell here. And it was shown in recent years that some types of astrocytes, not all of them, but some populations in particular areas of the brain, can also respond to an injury. And when they do that, they reactivate to a stem-like state. And after that, they can begin to form new neurons. And it is thought, again, that this is a form of repair within the brain in response to specific types of injury. So how does this all relate to glioblastoma? Well, I think it's very strongly linked, because we know that glioblastoma can arise from at least two different types of cells within the brain. One is the neural stem cells that I told you about a lot, and the other one are the astrocytes. And this mostly comes from studying mouse brains and mouse tumors. But we have some understanding that at least these two populations are important. And so over the last few years in my lab, we've been looking at astrocytes as a mutation. Now it becomes more plastic. And if this mutation is combined with an injury environment, could it be that these cells now, all of a sudden, anywhere in the brain, could give rise to a tumor? And so this is what this project really was trying to address. And to do that, we used mouse models. We do that for most of our work. And what we did is we did an experiment in which we took astrocytes within the brain, did not introduce any mutations. These are completely normal astrocytes, but we delivered an injury, and this is the cortex of the brain, where normally astrocytes do not form neural stem cells. And we compared this to a situation in which instead the astrocytes received a mutation that is known to be important in glioblastoma, again in the context of injury. And we then analyzed the brains 6 weeks after this injury to look at what would happen to the astrocytes. And what we could see is shown, represented in these images. You can hopefully appreciate that when the astrocytes are completely normal, after an injury they stay like astrocytes, so that they stellate cells, their name comes from star, and they remain completely normal. But if you have a mutation, only one, these astrocytes begin to change their shape, they retract their processes, and they begin to change. And this was happening quite frequently, and it was happening almost exclusively in the immediate vicinity of where the injury was delivered, suggesting that there was a synergy between the injury environment and a mutation. And this is alone was sufficient for the astrocytes to begin to change and perhaps be on their way to become neural stem cells, although at this stage they were not. So then we asked, okay, so what happens if we wait a little bit longer? Perhaps it's a slow process. And so we did the same experiment, but then we looked at the animals about a year later. And here, to our surprise, we saw that these cells actually progressed to a complete neural stem-like state. And this is shown here by a series of different markers that the cells begin to turn on. And not only that, but in some cases, they're able to form neurons, as you can maybe see from that cell on the bottom right, which has all these processes. And it's a cell that was an astrocyte, and then after an injury and a mutation, is now able to become a stem cell and go on to inflammation within all of our tissues, and this includes the brain. And inflammation can be really detected using stainings, like in these images. So we're looking for a specific cell type here, which is called the microglia, which is a cell that responds to injury by becoming activated. And so we looked at whether inflammation may be responsible for these astrocytes becoming stem cells as the mouse ages. And what we found, as shown in these graphs here, is that if you compare a young mouse that has received an injury, you see a lot of inflammation in the site immediately surrounding the injury, but not anywhere else. However, if you have an old mouse that has received the injury when it was young, and then you left the mouse to age, now you see that the injury is not resolved because immediately surrounding the injury there is still a lot of inflammation. But not only that, but the inflammation kind of expands so that surrounding the injury site you already still have inflammation that is persistent. And so what this suggests is that an inflammation that is driven by an injury early in life is then exacerbated by the inflammation that occurs as we all age and our brains become older. And so this is like a perfect storm that allows the level of injury to really transform the brain in such a way that it allows for these cells to begin to change their properties and become more stem-like. And we could show that this was the case because experimentally we could actually take a young mouse, injure it, and deliver a mutation in age. So this is a young mouse in which we deliver an injury and a mutation, and then a compound that increases inflammation within the brain. And this was sufficient for these astrocytes to now form these neural stem cells that had properties of early glioma cells. So this really suggests that injury has an important role in early tumorigenesis. And to sort of complete this work, we collaborated with an epidemiologist at UCL, and we asked them to look at whether there was a relationship between an injury in earlier life and the developments early in life, and the brain is intact and healthy, not much happens. The normal brain can actually suppress any sort of tumorigenic process, but as we age or if we experience an injury over time, the environment in which the cells reside changes in such a way that inflammation increases, and this is sufficient to allow these cells with mutation to begin to change and begin to form a tumor. So this study really gave us this first clue that injury and inflammation is really important. But the other big cell type that is really a culprit in glioblastoma is the neural stem cell, and it's probably the cell type that has been most studied and for which we have the most evidence. So what about neural stem cells? What happens to them and how do they give rise to a tumor? So to study this question, as I told you before, we cannot do it in patients. So we have developed over the years a series of mouse models in which we can deliver mutations to these neural stem cells within the mouse brain in the completely normal tissue, and then ask how are these mutations that are relevant to the human disease allowing these neural stem cells to change and form a tumor. And the beauty of this system is that we can also label the cells with fluorescent reporters, as shown here in red. So we make the cells red, we deliver the mutations, and then we ask how are they changing and how are they on their way becoming tumors. And so in this study that we published very recently that I will describe very briefly, we have asked exactly this question, and these are representative images that I hope you can see. Brain, and in this greeny turquoise, it's the white matter of the brain. Just for those of you who don't know what the white matter is, the white matter tracts are basically, they are made up of axons that are myelinated by those cells that I described at the beginning, the oligodendrocytes, and they crisscross the entire brain, and they're really like avenues of electrical conduction. So when we did these experiments and we tracked the tumor over time, so these are just images from different time points after we delivered the mutations, what was very clear was that early in the disease, the tumor cells do not go as. And so we probe this more directly, and of course one of the key features of tumor cells is that they divide uncontrollably. So we looked for proliferation markers, so markers that tell us that a cell is dividing. And we expected those to be higher in white matter, but actually this is not what we found. What we found is that the tumor cells divided to very similar degrees, regardless of where they were, throughout the tumor process, but there was a clear switch, whereby earlier in the disease, the tumor cells were not dividing very much, and then they switched to a highly dividing state, which is here, in which the tumors explode, and very quickly after that, the tumor kind of kills the animal. And so what that told us is that there are two phases to the disease, a latent phase, which is low, and this was very exciting because these tumor cells have all the mutations they need to form a tumor, but somehow they do not, and the normal tissue is keeping them in check. But then at some point, something changes, and this something changes specifically in the white matter tracts, so that now these tumor cells are able to go on to form a tumor. And so the question was, what is happening in the white matter and what are these processes. And so we took advantage of a really cool technique, which I won't describe in great detail, but it's a technique that allows you to look at what genes are on or off in a cell in space. So it preserves the sort of spatial information of where the cells are. And this is exciting because we needed to know what's special about white matter tracks. And when we do that and we look at what gene programs are turned on, injury responses that are being turned on. Not only there are injury responses, inflammations, there is mostly axons, and we know that when a brain is injured, for example, in a trauma, a car crash or something, the axons are probably the most vulnerable part of the brain. We can make our tumors in a mouse in which the neurons are labeled with a fluorescent protein, so we can study them in great detail, and when we do that, we see that the axons are indeed progressively lost, and you can see this with the green signal going down. But what the quantification showed us that the bulk of the axons are really lost at this critical transition when the tumor goes from the latent to the advanced stage, suggesting that this axonal loss program can really be a key driver of the tumor process. And the next key question was, if this is the case, how are these axons being damaged? Why are they beginning to disappear? And so we looked at this in more detail again, using a technique known as super resolution microscopy, in which we can look at cellular processes with great resolution. And what this told us is that what was happening is that the tumor cells are really mechanically compressing the axons, because we could often see these swellings here, and these are hallmarks of an axon being compressed, and basically the content of the axons are pushed on either side. And it's clearly happening where the tumor cells are, suggesting that there is a mechanical compression. So when the tumors is beginning to expand, it's injuring the axons, and this triggers this inflammatory response that is important for tumor growth. So why are we so interested in this? And the reason is that we know a lot about how axons die. This has been studied in great detail, initially by a scientist known as Waller, and the program that occurs after injury occurs to an axon is known as, and this occurs through a molecule known as SARM1. So when an axon is injured, the distal portion of the axon degenerates, and this is because SARM1 protein is activated. And what's really exciting about this is that we know that if SARM1 is inactivated, this program can be completely blocked. So if an axon is injured in a mouse in which SARM1 is deleted, now Wallerian degeneration no longer happens, and the axons remains intact, and in many cases functional. So it's a pathway that has sort of drawn a lot of attention as a potential way of tackling this because it's too much. So we asked this question. So we took mice in which SARM1 was completely normal, or mice in which SARM1 is deleted, and we generated tumors in both these backgrounds. And what we found was a very striking difference. So if we generate tumors in the absence of SARM1 and this Wallerian degeneration program, the tumors look very different. They're much less dense and much less aggressive. So you might appreciate that these ones have these holes, which are a readout of the core of the tumor dying and becoming necrotic and having new blood vessels being formed, but in the absence of SARM, this does not happen, and the tumors look much more benign. And so if that's the case, the question, of course, is, are these tumors less aggressive, and what happens to the mice, and how long do they live? And so we can measure these using these Kaplan-Meier plots that are shown here on the left, and you can see that in the absence of SARM, the mice live a lot longer compared to the controls, indicating that if we block this program of axonal degeneration, now we can extend the survival of the mice and make the disease less aggressive. But the key question was really, I told you that in the absence of SARM1, neurons can be preserved in a functional way. And at the very beginning, I mentioned that a key problem in the disease is that patients experience a series of cognitive problems. So if we are preserving neurons, is it possible that this intervention may also ameliorate this problem with cognition and motor function? And so we tested these in our mice, and we did this by measuring motor function through a series of tests. And you can see on the right here that if we do this in a completely normal mouse that has a late-stage tumor, indicating that by blocking this program, we might also preserve neurological function, which may have really important implications for patients. So just to summarize everything that I've shown you, it's clear that because it actually enables the tumor to progress. But I think what our work is beginning to show is that if we understand this injury program, we can harness them and target them in such a way that we can lock the disease in a more benign state and prevent its progression and the development of these cognitive problems. And so it suggests that there may be hope in the future for intervening earlier in this disease. And this is just a big thank you to my lab, and in particular the people involved in this work and my founders. Thank you so much for listening, and if you have any questions, please ask.
Thank you. Thank you, Simona. So, questions?
Thank you. Very interesting. A naive question. Do you think this is also relevant to recovering from surgery and treatment to avoid recurrence of the disease?
Yes, it's a great question. Yeah, we think very much so, because when the tumor is resected, obviously you will damage neurons and the tissue and axons will be lost. So something we would like to understand next is if we inhibit this pathway immediately after surgery, would that slow down the recurrence? And we would expect yes.
Thank you. Great talks, interesting results. Also from my side, maybe naive questions regarding the inflammations and the process of the disease. Autophagy or fast mimicking that may have some influence. You have studied this kind of aspects. Also, metabolic syndrome or NASH may have some effects on inflammation.
We have not, but you would expect those to be involved, but we have not looked. That'd be interesting. Yes.
Thank you.
Hi, thank you for the talk. I didn't get very well something about the mutations that you cited at the very beginning that targets or the neural stem cells or the
mutations that happen in patients. So we have a very good understanding, and it's many different ones, but it's a finite number, and they tend to occur in specific combinations. So in the mouse models that we create, we tend to take that into account, and we look at the sort of genome sequencing studies from patients, and we say, okay, these three mutations tend to occur together most frequently, and so we put them into the mouse to look at what happens. But it's a finite number, so there's often changes in EGFR. Another subgroup is PDGF. There are also losses of P53, but it's very heterogeneous. So even one patient will have some cells with one mutation and another group of cells with a different mutation. So it's a really complex disease.
Hi, fantastic talk. Yep. So another naive question, if you could comment about the difference using mouse models for astrocytes versus human.
Yes, obviously a human is not a mouse, so there will always be limitations. But I think the key problem is if you're really looking at the very early events and how normal cells respond to mutations, you're very limited in what you can do with patients. So in the context of the second story that I told you, we attempted to address that by looking at patient tissue in which there's been the same spatial transcriptomics technique done, looking at the margin of the tumor, which is more sparse, and it's sort of like evolutionary, probably the earlier stage of the disease, to see whether this same program occurred. And we found that, yes, we could see signatures of this program being activated. But you know, that's pretty much all you can do unless you go to a system in the lab in vitro in a culture dish with human cells, but then you don't have surrounding tissue and the immune system, so yeah.
Hi, thanks. Very interesting. So I may have missed this information. So you're proposing SARM1 as a promising target for cancer treatment. So do you know the level of expression of this protein in other organs? Imagine you have a drug one day, and then you get it goes in the body, and if you know if there is a problem, if you block the protein in other organs.
No, I mean, it's not selectively expressed in neurons, but it's most highly expressed there, and it's definitely much more common in neurons than other cell types, and it's expressed in the peripheral nerves as well, because they also undergo this program. But yeah, it's relatively good.
Okay, thank you.
Thank you. So I see there are other questions, but in the interest of time, I think we have to move on. Let's thank again Simona for this very interesting talk. Our next speaker.
Okay, am I audible? I think yes. So we have seen a lot of pictures of Michele's birthdays over the years, over yesterday. This one is a special one, I think, because this is the one where I think many of you sent videos, and we had a screening in the Lugano office. And then we have really group meeting, but group lunch. And this is one of the earliest things we had in during COVID in Genoa. And then it's the next birthday in Genoa. So I did my PhD with Michele around this special COVID and transition times. And then at some point I asked Michele what I should do for postdoc, and he recommended that I should do some applications now after all the enhanced sampling methods. So I moved to Frankfurt in the group of Professor Gerhard Hummer, and this is an atomistic simulation, and this is about 15,000 residues of protein. And this just explains the title of my talk, how do you make sense of this kind of molecular dynamics trajectories. So because of AlphaFold, cryo-EM, cryo-ET resolution, revolution, and so on, it's very easy now to simulate, or actually to get structures of this kind of large complexes, and this is actually one of the smaller systems. And here they wanted to look at the pre-pore and the pore. They resolved the structures back in 2014, and this is actually composed of 5 protomers of this kind. And each of these monomers that you see are about 2,516 residues. So this is the beginning of the problem that I faced during my postdoc. And so I thought, okay, we know a lot of different enhanced sampling techniques, and what happens if we start simulating something and maybe pull something, push something, bias something, and so on. So the first challenge that I faced was when you start using Plume for this kind of big systems, we have a problem with fit to template. So you cannot really put all the structure and use Plumed anymore. So I thought, okay, I still have to study how this pre-pore to pore transition happens. Are there any intermediates and so on and so forth? So how do I start? So I thought, okay, this is a pH-driven process. I could just start by protonating at different pH and run MD simulation. We had huge computing resources, and it's actually huge. So I could run microsecond long simulation of this 5.5 million atoms in a box. And then I ran this simulation, and you can see things are happening, but we don't understand anything. So you only see some jiggling and wiggling, and VMD does not even show you the secondary structure of the protein complex. So the problems that we encounter are huge. And as you can also see, this is a huge out residues that are creating differences in these different simulations. And then find out what can be mutated, run different simulation, find other ways to simulate the system. Today I'll not tell you about how I moved from this kind of simulations to the movie that I showed in my previous slides, but I'll focus on how I made sense of the simulations. So back in 1977, actually, I found this paper some time ago, and Charles Bennett actually wrote a nice comment. Even if speed of computation were not a problem, watching such a long movie would be. And this is actually one of the D. Shaw simulations of the small proteins, and if you can see, this is just 35 residues. We are way smaller here than even a single monomer. And when I look at them, we have 583 atoms, too many coordinates, more than 5,000 backbone distances, and then, so trajectories are high dimensional. We can run atomistic simulations, get microseconds of data, but then we have a lot of data, but we have no way to make sense of this thing that's going on. So we really need new techniques now, not only to simulation part is difficult, obviously, but we also need to understand how to analyze this kind of trajectories. So there is the need for some type of automation, but over the years, people have laid down very nice techniques. So
things that we already know, some dimensional reduction techniques, clustering trajectories, calculating free, a lot of things. There are already nice methods that do many of these things. And this first part requires a lot of physical and chemical intuition, even today, but we have exact methodologies for the second part. But what I really want to do now is I want to take my trajectory, put it into a sort of teapot, and magically get some nice reduced dimensional representation, clustering of the trajectory, or something that I can actually intuitively make sense of. And we need to know how does this pot cook. Well, feature distributed across your reduced dimensional space, kinetics-based clustering, and so on. So time scale of the metastable state, how much a state is, how long-lived a state is, and so on. Or once we know the states, or we have some labels for the state, we could get better reduced dimensions that are also interpretable. For now, I will just focus on this feature extraction part, and you will think like, okay, dimensional reduction already exists. What am I going to do here? So choosing CV is an art. Actually, it's also a science, but like Valerio also showed in the morning, you miss a single important variable and you miss a lot about your whole system. So in a way, when you are dealing with large systems, you cannot sit and look at every single angle. Rather, you need a way to process your entire set of distances, positions, coordination number, whatever set of variables you have at hand. We do have machine learning based collective variables, but you might need labels for your data to analyze them. Pratyush also has this nice method, Amino, that can take all your input descriptors and then cluster your descriptors and give you some data. However, when it comes to this large system, once again, so we have too many pairwise distances, and all of these methods become extremely expensive and processing gets really difficult. So when we are analyzing a molecular dynamics trajectory, we cannot, of this scale, we cannot just take all the high dimensional features and put it into trajectory analysis algorithms. So said, and passing it through untrained feed-forward neural network and getting a one-dimensional output from several different networks. So these are neural networks, and what are these going to do? Well, if we look at how this compression is working, so let's look at this simple data set. It's just X and Y in 3 different states. And when I pass it through networks like this, randomly initialized, and let's say I have 6 features, so you can have different depths and widths for your neural networks. So really randomize the whole combination process. Then we get lower dimensional representation if you take 2 of these vectors at a time, something of this sort. So this is basically more like an ensemble of suboptimal representation of lower dimensional projection. So people who are more aware of the machine learning training techniques, you might also know that other than, so some people, we often have a loss function that we optimize by taking derivatives of our loss function over training cycles and so on. But the other way to train a neural network is to get several representation of your output and then score them based on your loss function. So in some way, these are basically suboptimal representation of what ideal CV, one of them might be an ideal CV, but this is just a large set of suboptimal representations. Now, if this representation really works with a small system, so far we are looking at, let's say, alanine dipeptide, and alanine dipeptide has this 2 states. All of us kind of know that there are these 2 major states, and then we have the phi and psi coordinates. So if you see, if you put all of the 30 XYZ coordinates of alanine dipeptide, it makes a weird shaped transformation like this. However, some of the states looks like they're fairly well preserved. But we still have to evaluate if this compression is really working and if it actually preserves some important properties of our data set. So we need to try out different trajectory analysis tool to check if this compressed feature set is actually. But instead of putting all the 45 distances, if I take different numbers of compressed features, what happens is I can still recover some of the important time scales from this data set. So if you look here, the slowest time scale is recovered with very few number of descriptors, very few dimensional compressed space. But as we increase the number of compressed feature sets, we can also recover the the faster modes of the system. And if you look at the projections, the projection also looked slightly meaningful. So it seems at this point, this method is like, even if you compress the features, you can still recover some of the time scale and like kinetic properties of the data set. Then we thought, let's try some other method. So VampNet was promised to be one of the important clustering and kinetic analysis tools some years ago. And this method can take entire, very large set of features and then give you as output all the timescales and processes and different number of states. So we thought we should replicate this data and see what really happens. So in the paper, they used 666 inputs to the VampNet network and then optimized the model for 5 states. And they were able to get these 5 states with different population. So what I started to do is, instead of doing 666 inputs, I started with all the backbone distances. So I put the 6, 7, 8, 6 backbone distances into this VampNet network, asked for 5 states, and I got 4 different timescales. And I have plotted also to reference timescales for the same system. And it seems VampNet was working well also with all the backbone distances. Then I started to look at compressed feature sets as input. So I started with very few, so just 30 features, and get from any other method or from using all the backbone contacts for VampNet. So at this point, it seems compression does help by reducing the size of your network. However, it's kind of giving the same result. So why would you do something like that? And so you start to question, is there even a need for compressed features? So wait, I started to look at the protein clusters that I got, and these are the distance maps that I expected from the results that they published already in the original VampNet paper. And here, for example, this is a nice clustering, and these results are obtained using all backbone as input to VampNet. And I found that I could get 5 states, the correct populations and so on, but this happened only in about 3 out of 10 trials. So something was wrong, either with VampNet or when I used all of the features as input to VampNet. So I looked at the consistency of the clusters using this mutual information-based scores, and I used other scores too. And it turned out that it seems like as you increase the number of dimensionality of the compressed space, you do get more consistent clusters across different trials. However, with this, you have a 2-state splitting, and I did not understand what's really going on here. And so I looked at the population distribution. And here, I'm plotting the population distributions of the 5 states and for different trials. So either I have compressed features of different dimensions or all contacts. And from this, it looks like if you used compressed features at input to VampNet, you get more consistent results than using this large VampNet with all possible inputs. So structures are looking like they're more consistently clustered, and I'm using compressed features instead of VampNet with all the descriptors. Then I thought like the clustering measurement that I had could be skewed 5 different clusters, and each cluster has different protein structures. So I measured the fraction of native contacts of each of these clusters in each of the structures in a cluster, and then I take the mean of the value and I used it as a metric to look at the clustered consistency. Now here you can see when I plot the mean fraction of contacts across 50 trials, it seems with 30 dimensional compressed space, my results are like all over. So you can see there's a lot of scatter here. And when I use backbone contacts, I do get some consistency around here and in the bottom. However, here there are points that are missing and so on. So in some of the VampNet trials with all of the backbone contacts, I don't get all the clusters that I expect. So I started to look at higher dimensional compressed feature space, And I found that as you increase the number of features, you're up to, let's say, 100 or more, you start to get very consistent clustering results. And now if I look at each of these cluster representations, so if I take a single mean fraction of contact and I look at how the cluster really looks like, I see something of this sort, and you can see the sort of movement from folded to semi-folded, intermediate, and misfolded state as you go from top to bottom. And one important thing that I want to summarize from here is if you start using compressed functions as input to the neural network, you start getting more consistent result when you use compressed feature space. And it seems that you don't need too many features. If you use just about 100 or 150, it reports a different number of clusters and different state population. And these are like 2 recent papers. So in one of them, they reported that the folded state is about 68% of the population. In other, they found that it's about 23%. So there was a bit of confusion in the literature about the correct splitting of state, and depending on the kind of input features you used, the state split that you get differed. So we wanted to figure out if there is a correct splitting of some sense, and can compressed features give consistent results or more similar results to some descriptors than others. So first, I started looking at all possible dihedral angles of the system, all positions, all C-alpha contacts, all backbone contacts, and so on. And as you can see, with positions and contacts, we kind of get converged results with the slowest timescales. However, with dihedral, we get a bit lower results, the faster timescales. Now, if I look at compressed features, I see that the results lie somewhere in between. So the fastest process, it seems to be resolved very well with every type of input feature. However, when you look at faster processes, there is some diversity and confusion. So we started looking at the fraction of contacts again, and here I'm showing just a few examples, so a selected number of dimensions. Even though I asked VampNet to give me 4 states, when I look at the fraction of contacts, I can no longer find the consistency that I saw in the NTL9 case before. So the results seem to be scattered all over the place, and I can't see this plot and say how many states should be there. However, if I start drawing these lines and then maybe color the trajectory, color the clusters accordingly, I see some pattern emerging. And what I found is there seems to be 2 folded states, so folded 1 and folded 2, so this black one. And long story short, I could not get a consistent clustering with VampNet and any kind of input features for this data set. However, if I look at them, it seems there are definitely more than 4 clusters. However, if you start running VampNet with more than 4 structures, there are inconsistencies that arise and you no longer get VampNet crashes. Now, if I look at the population distribution of these clusters that I showed before, I can see that in some cases when we have 2 folded states, we have 2 states with about 30% population, and when the states are merged, the population are about 70%. And this kind of explains why standard MSM and graph VampNet were giving 2 different kind of splits. So our current conclusion from the Villain work is there is no proper clustering results that we can get from VampNet at this point. However, compression does help in exploring a lot of different clustering solutions, and this should guide you in some way to figure out what is the right number of clusters that you should look for. To make a conclusion from this, so there is a need for a complete package for analyzing large biomolecular trajectories at least, and compressed features offer a sort of fast and convenient way to compress a trajectory. You don't have to start looking at any important variables, and you can just put everything in and get something meaningful out of it. So with VampNets, we could get clusters and timescales, and also with TICA, we have seen the relaxation timescales are converged. So it seems compression does not kill all your information and does retain some of the important timescale and structural and density information that we should have in a lower dimensional space. Again, I'm calling it a compression and it's not clustering. However, although there are these limitations with random functions and compressed features at this point, I hope I could convince you that compression does really help, and it's a way to get feature extraction of some way. And this is going to be online soon. It's already on archive. And we have done some work also to cluster trajectory, so a new method that combines both density and kinetics to get better and more consistent clusters. And once we have the cluster, it's not enough, so I cannot go to my experimental collaborators with my cluster and say that this is it. I have to give them or suggest some mutations that are more biologically or chemically meaningful. And for that, we also have a dimensional reduction technique that takes our structures now, compares them, and gives you the highest ranking or most important features from that differentiates the clusters and so on. And with this, I would like to thank you for your attention. I would also like to thank Professor Humar and the Humar Lab for the fun discussions.
Thank you for the talk. Yes, questions?
I just want to anticipate the talk of Timothée Devergne, where he's presenting a method that might be of help to you. It's based on a diagonalization procedure that gets at the slowest decay mode of the system, so you can classify them according to their time and decide how many you need, and then you can represent the result of things and find out the cluster. How far you can scale this method, I don't know, but it would be interesting to see the two things.
The large scale directly from pH changing, this is actually not even feasible to see. So with pH, I see very minor differences in this. I don't see the swelling anymore. But then from this simulation, what I can do is I compare the interactions that are actually changing in this different simulation. And based on that, I mutate the system then to see the movie that I have in the first slide. So that's not directly from protonation changes. You don't see anything from there.
Thank you. Questions? So if there are no further questions, we thank again Yashrita. So now we have a coffee break.
Tu adesso proprio sei in Svizzera, no? Tu sei in Svizzera. Quando è che sei? Sono ancora affiliato in Ah, fai avanti e indietro? Ma per la famiglia? Qui non ci vedo.
Okay, so welcome back. The first speaker of this second part of the afternoon session is Christophe Del Lago from University of Vienna. Thank you.
Thank you very much. Well, thank you, and thank you for having me here at this wonderful celebration of Michele's birthday. I'm not really part of the family because I never worked in Michele's lab, but I feel that this community has adopted me. And that's very nice. It's a good community to be adopted by. So I'm going to talk about some work, Bastian Falkner at the University of Vienna and a postdoc, Alessandro Curetti, and also Phil Geisler, who was involved in the first stages of this work. Of course, Phil was a dear friend of many here. He died tragically now 3 years ago. Phil really was a wonderful scientist, a great mentor, and PS was the dissociation of water in liquid water. So the problem is to understand how a water molecule dissociates. And in water, this happens rarely. A water molecule has a lifetime of about 10 hours. So in order to understand what's going on, Phil was sent over to Stuttgart to the lab of Michele, and with the help of the people there, he did some transition path sampling simulations. And this is this sequence here of pictures shows roughly what's going on. The dissociation of a water molecule happens along a wire of hydrogen bonds. So here you see that, and this happens on the femtosecond timescale. So the trajectories that we collected were 150 femtoseconds long. The two ions that form separated along this wire, and if the wire then breaks, the two ions cannot recombine quickly, and they have a chance to separate and move away from each other. Now, at that time, these were very expensive calculations. This was done, of course, without machine-learned potentials with ab initio. So we collected about 10 independent trajectories. That's what we estimated by looking at correlation. So that's not much, but we tried to make sense of these trajectories. Ali has in equilibrium is of course the same. Now, at that time we really couldn't do what we would do today, namely an analysis based on committers to understand the mechanism. To give you an idea what that means, let's look at another example, another system that we studied at this time. Pratyush has already mentioned it. It's the dissociation of two ions in liquid water. If you action coordinate, then you would expect that the interionic distance determines the committer, and the committer is the probability associated with a certain configuration that if you start from there, you go to one of the stable states rather than the other. Now, if you collected configurations corresponding to the top of the barrier, you would expect that all of them have a committer of 1/2 if this is a good reaction coordinate. But this is not what happens. You find that there are two values of the committer that appear, 0 and 1, and this is because you have a situation where you have neglected some degree of freedom, and you collect configurations from here. They have a committer of 0, or configurations from here, and they have a committer of 1. So this is a clear indication that something is missing, and and the thing that is missing is a reorganization of the water molecules in the first solvation shell, of the ions. Now this idea of the committer, very useful, is not a new one. Already Onsager thought about the committer when he studied the recombination or dissociation of ions. So he studied this very simple process where you have two charges. They are initially at a certain distance under the influence of a Coulombic attraction. Now, depending on the distance, the two charges have a certain chance to recombine or to finally dissociate. And this probability is nothing else than the committer, and Onsager realized that the committer has to satisfy the backwards Kolmogorov equation, which is written down here with the boundary conditions that it is 0 at 0 distance and 1 at infinity. And this is an equation that you can solve the probability of 1/2 for the two ions to dissociate. Now the committer is now the basis for a lot of methods. It's the basis for transition state, transition path theory, and also of a new method based on the committer, because in a very Parnellian way, Michele and collaborators realized that you can write the solution of this equation in a variational way. So the committer that minimizes this functional here is the true committer, and then they came up with a particular way to sample in order to sample efficiently the region where the committer changes. And so this is a way to compute the committer without calculating trajectories. Now, actually, I would be very interested in knowing if anybody knows here if there is an older reference where somebody calculates the committer based on the backwards Kolmogorov equation. This is the first one that I found. But there is an older reference to the committer, and this is the so-called problem of points. This goes back to the 1500s. The question is the following. Imagine you play a game of tossing a coin. Two people play this game, and they agree that the first one that award be split between the two people. Now there is a certain Luca Pacioli, who apparently is the father of accounting. He invented accounting, and he was a friend of Leonardo da Vinci. And he said, well, it's clear what you need to do is to, and he said, no, you cannot do that because, a ratio of 2:1 is completely different if you have it at the beginning of the game or at the end of the game. If towards the end of the game, one is ahead by a ratio of 2:1, you will never be able to catch him. But if it happens at the beginning, easy. Right? But this was not the right expression either. Cardano, famous mathematician, also had a suggestion, not the right either. The people who really solved the problem were Blaise Pascal and Pierre de Fermat. And they realized what you need to calculate is the expectation of the probability. Imagine that you get to a certain point here in this space of the game, and that player A has won A times, player B has won B times. What's the probability for player B to win? To get to this site rather than to A. All you need to do is to count the different pathways that exist for player B to win. And this is what Pascal did, obtaining this expression. And if you plot it, you find this, namely that the probability for player B to win is 1/2 along the diagonal, of course. Because of symmetry, you expect that. And then you have this blue region here where it is 1, basically, and this red region where it is 0. So this is a committer, right? And the transition state here for this reaction is the white line. Is the white line here. Right? So using the committer, you can determine whether a reaction coordinate is good or not. And that's often not so obvious. In this situation here, for instance, you would think, okay, Q is a good reaction coordinate because it distinguishes the two stable states. And there is a nice barrier in between these two stable states. But of course, looking at this, it's clear that in order to go from A to B, you have to pass through this transition state over here. And Q prime definitely needs to be part of the reaction coordinate with a gate here. Right? But the cow is not alone. There is a little fly here. So we look at the system cow plus fly. The fly likes to be near the cow for obvious reasons. If you go to the mountains, you know that. And then you ask, so how can I move the system cow plus fly to the other side? Right? So if you move the cow, the fly will follow. And both of them are on the other side. But if you try to do the same thing by moving the fly, you will not have much success because the fly will just fly back to the cow. So here you clearly see that the cow is the reaction coordinate. You might call it the reaction coordinate, haha. and the fly, although you can use it to detect where the cow is, that will not work as a reaction coordinate. And this is something that, of course, these two cowboys here, they knew it perfectly, you know. In the mountains where I'm from, actually, that's probably in Italy as far away as you can get from Sicily, in the mountains where I'm from. But, you know, I think that mountain people and island people have some similarities. We can talk about that later. Anyway, so they, in the fall, basically about now, they decorate the cows when they bring them down from the pastures at high altitude down in the valleys. And these two cowboys knew exactly that they had to drive the reaction coordinate with these Gaussian bells here to bring them down. Just, you know, taking the flies wouldn't have done the job. When we worked on transition path sampling almost 30 years ago now, knowing the transition, knowing the reaction coordinate is exactly something that we wanted to avoid. Sequential method is affected by correlations because the new trajectory will be similar to the old one. And you have to generate a few ones before you get a statistically independent one. In some cases, you have to wait for a long time. So this is a little movie that Sebastian Falkner made for his thesis defense. You have a transition from A to B here, and there is a no-fly zone here in between where the system cannot go. Now, the system can go from A to B through this channel or down through this channel, and you see here that it takes a long time before this simple Brownian motion here manages to find the other channel. Now, the question is, are there ways to reduce these correlations? Because these correlations are, of course, expensive. If you need to generate many trajectories in order to get an independent one, that costs. You would like to avoid that cost. And one way to think about this is in terms of machine learning. So one way to avoid correlations that has been proposed for the sampling of configuration are generative models like normalizing flows. Here the idea is that you start from an easy-to-sample distribution, and then you learn a transformation that transforms to the distribution that you want to sample. I have what Frank Noé called the Boltzmann generator. You can generate configurations in a one-shot way without correlations, because you are able to sample this simple distribution in a correlation-free way. So also the complicated distribution is correlation-free. A simple distribution of pathways like a random walk, and then we transform it to the distribution that we want, the transition path ensemble. If you try to do that, good luck, because these normalizing flows have a problem with scaling. They don't scale so well, meaning that the errors accumulate if you go to higher dimensions, which makes the reweighting at some point impossible. But you don't need to do it that way. You could do something less ambitious and generate shooting points from which you generate trajectories with regular molecular dynamics. Right? So here the idea is to start from a easy-to-sample distribution, let's say the Gaussian distribution, generate shooting points, shoot off trajectories, and then reweight the ensemble of trajectories that you have produced. You need to do that because the normalizing flow is not perfect. This works with methods very similar, basically to a Boltzmann generator, but with a bias, because you need to produce your shooting points in regions where they have a chance to lead to reactive trajectory, and there is a catch, right? Because you don't really know where to put these points. But if you knew, then you can you can actually produce trajectories very easily, and they are uncorrelated. So here we did that for a very simple two-dimensional model, or here for something a little bit more complicated, a configurational transition for a simple 2D polymer, and also here the generative model produces, converges much quicker. But the problem, of course, well, you have two problems. One is the scaling of the normalizing flow, but Pratyush tells me that the neural network to predict the committer, and this is exactly what we need. Right? We need to know the committer, because then we can bias our generative model to produce configurations in the region where the committer is one half. Now, committer learning can be done. So here we have done it in the context of transition path sampling of the AIMP method, together with Gerhard Hummer, Roberto Covino, and Hendrik Jung. What you see here is the learned committer versus the sampled committer. This model actually takes in some descriptors, I don't know, a list of 20, 30 descriptors, and then predicts the committer based on these descriptors. You can then later do an analysis to find out which of these descriptors really are important and carry the information. Then you can use this in an on-the-fly way to learn the committer, and at the same time then use the learned committer to bias your generative sampling of the shooting points in a way that you get trajectories that are reactive. So this is something that we have just started doing. It's the master thesis of a talented student, Max Negetli, and we are now working on applying this to more complex systems than just a 2D potential. I would like to spend I still have a couple of minutes, right? I would like to spend these minutes to talk about another method that is not based on machine learning, but has the same goal of reducing the statistical correlations between trajectories. And here in this method, which we call the shooting point exchange, the idea is to combine transition path sampling with an enhanced sampling method like metadynamics, and to sample an extended ensemble which consists of the path sampling simulation, and periodically there are exchanges between the path and the configuration. These exchanges are done in a way such that you pick a point along the path. This is the new configuration, and you shoot off a trajectory from the configuration, and then you switch. Now, for 2D model, this works very nicely. You can reduce the correlations a lot. What's plotted here is the error in estimating the fraction of trajectories that go through the upper channel. It needs to be 1/2. If you do it numerically, you make an error, and this error decreases like that for shooting point exchange and like that for standard TPS. Of course, standard TPS is hindered by this barrier that separates the two channels. But this method can be applied to more complex systems. Like here we studied the isomerization of proline in a tetrapeptide. What you see here is this tetrapeptide the water molecule. It's immersed in explicit water, 40,000 water molecules that are not shown here. And here you see this transition where this oxygen moves over to the other side. Now, this transition can happen in basically 4 different ways that are, because the oxygen can move clockwise or anti-clockwise, and also this, this region around the nitrogen can deviate from planarity in 2 different ways. So there are 4 channels, and the question is, can all these channels be sampled? And if you do it with standard TPS, the answer is no, because TPS has a very hard time to go from the clockwise to the counterclockwise mechanism. For Michele already knows about it. The scientific advisory board of the Erwin Schrödinger Institute in Vienna has decided to award the AZ Medal 2025 to Michele. And you will come in November to Vienna to receive your prize. It's actually a medal made of silver from a company in Tyrol, because there is a long tradition of silver mining in Tyrol. Actually, they were the first victims of globalization, because when they discovered America, then the silver prices in Europe went down. And but there are still some companies around, and one of them made this medal that shows Erwin Schrödinger here in a pensive mood. You see, mountains and islands are similar.
There were mines of sulfur,
right.
Very profitable. And then the damn American found the sulfur, and the way the quality of the was such that they could extract it very easily and cheaply, and that was the end.
Yes, yes. It's always the Americans. Yes, that's the end. Thank you.
For this beautiful talk.
I want to underline a property of committer, which can be extracted, of course. It's this probabilistic nature of the thing that allows, there is a famous result of yours, that if you look at alanine dipeptide, the angle phi is the order parameter, but the transition state is, for the transition state, it's also important the theta angle. And by looking at the committer in that way, we were able to recover your classical result. That's all.
Well, I agree.
The questions? I had a question about the computational cost of the method that you have shown, that you are applying it already on a smallish but realistic system. So can you apply it to much larger and complicated system? What would be the computational cost?
So in the calculations that we did here, the computational cost of the metadynamics simulation is much less than the transition path sampling, right? So as long as you can calculate trajectories, I think it really depends on...
Would they have to be continuous trajectories?
Continuous in which sense?
A complicated system where the transition is diffusive, would be still...
Well, I just wanted to say, it really depends on how long the transition time is, right? If it's diffusive and it takes, and you're not able to do the whole complete transition in your, molecular dynamics, then you're out of luck, of course. Right? But you need to be able to generate a number of trajectories. So that's
Would it make sense to combine it to methods that approach this problem, like transition interface sampling? Or
Sure, but transition interface sampling is affected by the same problem. in a sense. So, but of course.
You have this picture where the pass has to find the other way around, right? And you say there are correlation times in there. Is there a separate correlation time to find the other way around? It's not the same as the transition.
No, I mean, that's the, so this correlation measures how many attempts you need to find the other transition, so the other pathway. In this example, I think you will never be able to even determine this correlation time because you cannot run enough trajectories. If you don't couple it with metadynamics, just with regular TPS, you will not be able to this is what I meant by correlations.
But there is a statistical mechanical expression for this time.
You can write one down. But this is not the correlation time along a trajectory, but the correlation time between trajectories.
Regarding your question of if you know somebody who did this already, you have to read Russian papers, right?
Yes, thank you.
Okay, with this, we thank you.
Thank Francisco for the introduction and the organizer for inviting me to this meeting. So so I started, let's say, around 3 years ago or 4 years ago at the University of North Texas, where my group is kind of working on different problems within, let's say, biochemistry and material science. So application-wise, we focus mostly on, let's say, biomolecular system like protein-protein interactions, targeted protein degradation, and molecular crystals. And we also do a bit of method development. So here, what I'm going to talk about is more like applications in the area of protein-protein interaction that can lead to diseases. And often what we see that, for example, mutation, they generally affect the off rate more than the on rate. And I'll talk more about that. Also, it's kind of important how we can use small molecules to kind of modulate protein-protein interactions, for example, stabilizing them, using so-called molecular glues, or maybe destabilizing them, having some inhibitors that inhibit interactions. And also, protein-protein interactions are important in various therapeutic modalities like targeted protein degradation, antibodies, and so on. So it is a critical task to kind of predict on binding kinetics and pathways and so on using atomistic simulations. To say one maybe trend that is known generally for protein-protein interactions is this one here. It's a paper from like 10 years ago, or like 12 years ago, where they looked at correlation between binding affinity and some properties of, let's say, protein-protein interactions. One correlation that they found, although maybe weak when you look at this graph, is that there's a correlation between the binding affinity and the amount of surface area, basically the area of interaction between the two proteins. Although you can see it's not really great correlation is here. But also one thing that they did notice, there is no obvious relationship between the nature of the interactions, like if it's hydrophobic interactions or electrostatic interactions and so on. Okay. So say, of course, let's say, just remind everyone about the importance of like kinetics in biomolecular interactions. Affinity, like the frequency difference between the bound state and the unbound state. Of course, we can also look at the, let's say, the off rate, which is, let's say, how fast the ligand binds, or the on rate. And normally, when we talk about the off rate, we talk about the residence time and so on. And of course, everything is related to this equation here. So why should we care about kind of the off rate or the binding rate? This was kind of established like some years ago, like 20 years ago, by Robert Copeland. There's a time scale issue. Like if you look at, let's say, the time scale we can access to normal atomistic simulations, they're limited to a microsecond, while normally we look at maybe much higher binding times. Just one example, like this system here, we'll talk about later, binding per star, where it has an unbinding time of, let's say, 24 hours, a day. So here, or like a half a second. So of course, like this one. And this is also an interesting graph. This is something that's extracted from a database of like 80 like protein interactions. So this is like 80 kF values to a histogram. And you can see here like the shortest unbinding time on order of seconds, and the largest one is like a half a year, like a 1/3 of a year. So you can see here that unbinding of protein interactions is really rare events. To say we need to have some enhanced sampling method to comparison to that. So say I, of course, come from the group of Michele, so the areas which we can reuse is, of course, in this area here, or collective variables-based methods. So this is a review paper, which I wrote a few years ago with people in the field. We're going to try to classify different methods, and to say, and I, of course, use here, like when people have discussed, like collective variables-based methods. And of course, one thing that they came access to is kinetic rates. Dhiman already mentioned this earlier, but I need to kind of go over it again. So normally what we use is infrequent dynamics, where the main assumptions is that like infrequent dynamics was developed by Pratius and Tivari, Pratius and Michele, and also the main assumption is that we have good collective variables that describe the rare physical process, and we deposit no bias on the transition state. So what we can do, we can actually test the quality results by doing this kind of p-value test or Kormorov-Smirnov test, and so on. And of course, nowadays, we have various methods to build the bias in frequency dynamics, Weyer's plotting, frequency adaptive dynamics, and OPS plotting, like we discussed earlier. And now here we use like this hyperdynamic rescaling formulation to rescale the time, but of course, nowadays, there are also other methods to get this time here. Of all the papers that look at protein ligand and binding. As you can see, there's a lot of methods, a lot of different methods, a lot of different systems, and so on. There's a lot of interest in this field here. So this is protein ligand. So the same paper, they also look at they listed, let's say, application to protein peptide and protein-protein interactions. And you can see here, there are much fewer applications. There are basically like 2 systems or 3 systems people have looked at. So when I saw this, I decided this was kind of a gap in the field where I want to focus my group on addressing, so on. Yeah. So like we want to kind of address this and to kind of apply this kind of kinetic methods to protein-protein interactions. Okay, so just say we're starting this, so I'm going to talk about some projects that we're working on. So say some research questions that we are working on related to protein-protein interactions. For example, we're looking at protein peptide unbinding, which we're also looking at the effect of protein force field, the water model and so on, effect of mutations, and also what we want to go towards is like looking at the effect of stabilizing molecules. What do you have like molecular glues, something like that affect the binding. And one area that we're starting to work on is so-called targeted protein degradation, where we have like a molecule in between that links two proteins together. The area you have to look at the effect of cooperativity and so on. Okay, so the one project that we're kind of finishing up and I want to talk about here is to look at protein peptide unbinding kinetics. In particular, we want to look at the effect of protein in water model, like the force field. Because you're able here, people are using different methods, but different force fields, so it's very hard to distinguish what is the effect of the force field and what's the effect of the method. So I kind of want to understand like what is the effect of the force field when doing these calculations. So as I say, results that we obtain. So now we're going to focus on protein peptide systems where we know the binding modes, like we know the crystal structure, but maybe in future we go towards like design of peptides and so on. So this is all work done by my student Jai and Aloka. So here we're working on 2 different systems. One is SH3 SOS peptide here, and this system here has an unbinding time around 100 microseconds. This is mostly driven by electrostatic interactions. There is this kind of arginine residue here, while that is charged and has like electrostatic interactions. This is MDM2 P53, a well-known system, a protein peptide system, often used for testing, and this has an unbinding time of half a second. But it's interesting here that this is, let's say, driven by hydrophobic interactions. This is kind of a choice. We decided to like have one system driven by electrostatic interactions and one system driven by hydrophobic interactions to understand like how these different things behave. To say, we want to understand how the choice of the force field affects the unbinding kinetics. So say there is no published study addressing this so far as we know. Okay, of course, we all know we need to select the collective variables. So here, we basically look at, I would say, pretty simple chemical intuition-driven collective variables. So in both systems, we were using 3 collective variables. One is basically we select the residue at the interface and look at the center of mass distance between them, like the center of mass distance between the interfaces. And then we select important residues on the peptide and look at the coordination of that residues with the heavy atoms on the larger protein, something like here. So here, like this protein here has adenine residue, a valine residue, while everything else is basically a proline residue. Here then we have, let's say this one here, has like these three hydrophobic residues, and we use some combination of these coordination numbers here. So now, certainly, you see here, so here are the three different collective variables. So this is the distance between these residues. So this is one of the biased CVs, and this is the coordination number. And here we also look at the fraction of native contacts. Just as a control, we don't bias this collective variable. And if you start to bias the system, you can start to see actually that there are no binds around here. You see that the coordination goes to 0, the native contact goes to 0, and also the distance increases and so on. So we can assign on binding event here. Now, what we go to do, this is the simulation time, roughly, let's say, 400 nanoseconds. So then what we do, we can do the time rescaling according to this equation here from like hyperdynamics formalism. And now we have a rescale time here in milliseconds, around, let's say, 70 milliseconds. And here we can see we start to be in the order of the time scale of the system, and so on. Okay. And what we do, of course, we perform multiple simulations, at least 20 simulations for each force field. And this allows us to get a time distribution that we know should follow a Poisson distribution, the 6-modal. And we do this current p-value test. And what we also do is that we estimate like a 95% confidence interval by doing a bootstrap analysis. So always we show results like here, where we kind of are doing this bootstrap analysis to get like an error bar for the results. So when I show the results, the cross here would be like the result directly from like the 20 runs, while this dot here is the average, and then there's the error bar, average for the bootstrap analysis. So let's get down into results. So here is, let's say, we started with first some commonly used force fields, like Ampere 99 SB ILDN using tip 3P, Ampere 14 tip 3P, Ampere 19 SB, Charm 36M, which is, let's say, the latest one on Charm, is much lower. So here we can already see that, like with the latest force fields used in the field, we can already see like they're on the opposite side of this kind of range here, like in terms of time. This is much faster on binding, this is much slower on binding. And we don't have the Ampere 99, Ampere 19 data point here, but initial result indicated is also a similar result here. Well, here we actually see a bit more difference between the two force fields. So we can then start to see that the behavior of the force field is different between the two systems, which might indicate, let's say, due to the nature of the interactions. Okay, and so on. Then we also tested the water models, which is of course an important thing. So we started to use, let's say, TIP4P 2005, which is like a well-known 4-point water model. And in this case here, both using Ampere 99 and Ampere 14, they're pretty similar kind of in the error bars in both cases. Well, here there's a slight difference here. But here they're pretty similar. But still, let's say, the water model doesn't seem to be a big effect, unless we go to this water model here. This is called TIP4PD. This is a water model developed by Paolo Bustelli and Stefano Piana at the DSO group. This was a water model that is designed for, let's say, intrinsically disordered peptides or proteins. So it's supposed to give a better description. And one feature of that water model is that they increase the dispersion interaction between the protein and the water. So you can see here, that gives considerably lower unbinding time than anything else. So my kind of hypothesis for that is most likely that, let's say, when the peptide starts to unbind, the interaction between the water and the proteins is much stronger, so the water just comes quickly in and rips this peptide away. So I would think most likely this is here is probably an unbalanced description for this one. We kind of followed up on this kind of route, let's say, so yeah. So we also followed, let's say, up on this kind of route of looking at further force field developed by the DSO group, and so on. And let's say there's Ampere, is a force field developed, let's say, for protein-protein interaction. So we're quite curious to see how that performs, because it's designed to give a balanced description, let's say, folded glucoproteins, ITPs, and protein-protein interactions. But interestingly, that one, let's say, gives quite low unbinding time compared to everything else. And particularly here, for this system here, we even saw unbinding in an unbiased simulation, showing that much weaker bound, and so on. And also, yeah. But there's some technical details here. For example, in this amber, they actually scale the charges by 0.9. So that one could have an effect. So we tried a version of the force field that they developed where they don't scale the charges. And you can see here, for this system here, it's a quite big of a difference, most likely because this is driven by electrostatic interactions and so on. Okay. So, yeah. So overall, to say, it's kind of maybe not a nice picture to see, but overall we see that there's kind of a 2 orders of magnitude difference between the two different force fields. We can see here, going from the highest unbinding time to the lowest, there's mostly 2 orders of magnitude. So we can see clearly that the force field has a big effect. So on. One thing that we try to look at is like if you can correlate the unbinding with the bound state, the property of the bound state, if you can see something there, and we see that we cannot have any correlation about the trends compared to the bound state. And most likely this is due to the fact that this force field, they're more modeling the transition state, or more likely they're modeling both the transition state and the bound state. So it's hard to distinguish this. Like for you to do, maybe use some methods to identify character of the transition state to further understand the results. And also, one can look at the binding affinities, because that will give a different insight, and we'll start to do in hydrophobic, and so on. And to say, I find it quite curious and kind of that this amber, which is designed for protein-protein interaction, give considerably lower unbinding times. As I say, it looks like we have this kind of towards a magnitude difference. So my personal opinion is that like we aim for, let's say, absolute experimental comparison is not really the way to go. So, yeah. And one maybe thing one could look at might be to see, let's say, what about polarizable force field? They might maybe, like, be curious to see how they perform. Could they fix some issues here? Okay, so in the last 5 minutes, I'm going to talk about another system, which is, I think, results which I'm pretty excited about and pretty impressive, is to look at the effect of mutations. To say, we want to look at, let's say, relative things. So what about the effect of mutations? So the system that we're going to look at is a well-known system called Barney-Bar star, and has a very strong electrostatic binding. And the unbinding time is around 18 to 35 hours. So pretty strong bound. And if you look at the system, the interface, you have a lot of this charged residue of the interface, like aspartame 39, aspartame 35, and so on. So these are all important residues of the system, like of the binding. So if you look at mutation experiments, this is something which I found very impressive when I saw this first. So here you have the wild type, which is in this case like 24 hours or something like that. You make 1 mutation. You mutate aspartame 39 to alanine, and it goes to 1 second. Just 1 mutation goes down by 5 orders of magnitude. So it's a huge effect of, let's say, just mutating this 1 residue. If you mutate a certain histidine, you will have, let's say, like something, if you mutate both the histidine and aspartame, it goes to kind of below 1 second. So as you see, it's a huge effect of the mutation. For some technical reasons, we are not looking at the wild type, we are looking at the pseudo-wild type, 2.4 hours. Still quite long. And still we can see the same effect, go like you do this 1 mutation, go from, let's say, 2.4 hours to, let's say, 1 second. So the question that we want to see here, can we kind of predict relative kinetic rates between mutants? So this is, let's say, work done by my student in PAS. So first, we looked at the pseudo-wild type. And here we're already pretty impressive. We get basically the pseudo-wild type pretty close, or very close, within the error bar. So here we get the experiment at 2.4, what we get from infrequent meta-analysis is like 1.4 to 8.9 hours, which is pretty good. So there's not any papers that really looked at this system. There's one paper by Frank Noe from like 8 years ago, using Markov state modeling with the same force field. But if you really look at the paper, their unbinding time reported has a huge error bar, basically from like 10 seconds to 94 hours, so it's kind of impossible to say something like that. So now, let's say, what about the mutant? So what if you mutate this aspartame 39 to alanine? And that is something which, let's say, this is a good start. I'll look at the absolute number, but let's say we want to look at the relative things. So now we do look at this mutation. You see here, so this is like the result. We do this mutation, the experimental time is 2.1 seconds. So we're getting, let's say, around 1 second. So we capture really the effect of the mutation here. And you can look at this, meaning this is pretty impressive here, like the difference between the wild type and the mutant. So we look at, let's say, intermediate residue, this aspartame 39, has an off time of like 0.6 minutes. We're still a bit higher here, but still within order of magnitude. So this is probably not too bad here to say to this mutation. So that's, let's say, future work that we're calling. And also, I'm going to skip that, we have time. So finally, I just want to say, hamingju með afmælið, Michele, happy birthday. I spent many wonderful years in Lugano. It was a great time. And I think also this was a good time. Some of us who are here remember this time. We went to this Villa Garibaldi for a group retreat in 2013. It was a very good retreat. And I do remember the food was very good. So I'm going to share one story of Michele. Okay, so I think it was about the time when I was applying for a postdoc with Michele. I interviewed Michele. I think a couple of weeks later I was kind of anxious about like what was going on. Like I interviewed Michele, I hadn't heard anything back. I was a bit anxious, looking for postdocs. And I sent Michele an email. And he responds with a single sentence with this line here. Things are looking good. And I told Michele before, and like, I don't know how I should interpret that. But then a few days later, Daniel Weir sent me a contract and so on. So I think Michele, things went pretty well, pretty good. But I say, yeah. That, I just want to thank you for your attention and open up questions and so on.
Thank you. So, questions? Maybe I start. So, it's extremely interesting what you have done looking at all this force field, but it seems pretty hopeless. So, so you think it's a problem with the superficial charges on the protein, or, I mean, we just need polarizable force fields?
Meaning there could be a few things, like maybe polarizable force field is one thing to look at. That is something which it's more difficult to find code that support it and so on, and like it will take a bit more effort, and like the time, maybe systematic in a sense, like it might depend on like type of interaction and so on. It's maybe, I want to say, it's not always the same trends for each system.
What about the ions? Have you used the same ions that they have in the
So, AMP99 uses, I don't remember the name, but AMP99 and AMP99 uses different ions than AMP14 and AMP19. So, like, we always use the ions.
Same concentration as in the experiments in this.
I'm not totally sure. Yeah, we have 0.15 molar. 0.15 molar. I showed here we have tried some effect of the ions, and that seems to be the effect of the ions. So the unbinding seems to be minimal actually. But as I say, the ions are like just the same as used in the force field. So like AMP99 versus AMP19 uses different set of ions.
Thank you, Omar, for the talk. I really enjoyed it. Water plays a very important role in this process, and there has been significant progress in in modeling water and making good potentials. But this doesn't seem to be sort of percolating to this type of problem. How do you see the future of that?
classical rigid 3 or 4 body water models. That's like what it limits using, like if you use it with this, let's say, force fields. Meaning, to say, here there's not a big difference between the water models. We would like to add OPC here, just to understand the effect.
Our next speaker is...
Okay, normally I don't need a microphone. I'm too loud and neurotic anyway, but I'll try to make do and not scare you off while using a microphone. And thank you, Ali, for inviting me here. I'm extremely excited to be here. It's my first time at ICTP. It's a really special place, I can tell. So thank you so much and all the other organizers. And I'll say something about this in a second, but I want to first say to Michele, Yom Holedet Sameach, happy birthday. And thank you for having me here for the special 80th celebration. I had only 2 years with you in Lugano, which is a short time, but they were some of the most meaningful years in my professional career. And when I came for a short visit before I started in the group in Lugano, I met you for maybe 30 minutes, and we chatted. And the only thing I could think about when I came out of that meeting is you thought about scientific problems in a completely different way than how I thought about problems. All I wanted to do was to learn from you how you think about scientific problems. So I hope I managed to do this a little bit, and thank you for the opportunity of doing that. You also created an unbelievably talented environment in your group and intellectually stimulating with. And not only that, with people that I believe I made friends for a lifetime in just 2 years. So thank you so much for doing this. And this is a really cool picture up here. Michele, do you remember this? We went to Trento. I was working on bosons. And Michele liked the results. So immediately he called up Stringari and organized the visit. And we went to Trento and met Lev Pitayevsky, of having him there. And by that time, I already also had a daughter. So I got to meet a side of Michele that I had no idea existed. And so Nono Michele is how he introduced himself to my daughter Noga, who's now much bigger than this, but I'll cherish these pictures forever, and there she is bossing him to give her a strawberry, which is perfectly accurate for that age. So thank you again, Michele, for everything. We worked together on a topic I never imagined I would work on, which was path integral molecular dynamics for indistinguishable particles. And we wrote some really nice papers in black at the top, which I'm super, super proud of. And I worked with these wonderful people, Valerio, who's here in the audience, and Chang Wu and Michele Invernizzi, who are not here. And I wanted just, before I tell you about my main talk today, I wanted to give you a quick update on what we've been doing with this, because I think you will like this. The torch has been carried by my two brilliant students, Yotam Feldman, my postdoc, and Jacob Chiger, my MSc student. And it even got a Best Paper Award from JCP a couple of years ago. And the problem, I learned from Michele that if you work on hard problems, whatever you do will end up being interesting. So the problem he gave me when I just started, again, we talked about doing completely other stuff. And then the first day I come in, he gives me this problem. I mean, we did talk about path integral stuff, but not particles. And he told me maybe we'll do this for fermions and bosons. Fermions is even harder. And the problem, I think all of you are familiar with what path integral MD is, but in case you're not, I'll tell you everything you need to know about it in one sentence. And it turns out that every quantum system could be mapped onto a classical system of ring polymers that has exactly the same partition function. So if you use molecular dynamics or Monte Carlo simulations to sample the classical ring polymer system, you get quantum expectation values. And the first time I heard this is such a hard problem, because for n particles, there's n factorial permutations, okay? And you can think about these polymers like this. You can also think about them as paths in imaginary time, which is what I will mention in a second. And so I never imagined working on an exponential scaling problem before I went to Michele. And we did some nice work together, because if you try to do these simulations as a function of the number of particles, naively, in PIMD, you get the blue curves. So if you follow it, you see that basically you can't run any simulations for more than 5 particles. It's hopeless completely with PIMD. And then Michele and I and Valerio, we developed an algorithm and we showed that you can actually evaluate the bosonic spring ring polymer potential recursively exactly with no approximations, and that scales only cubically with the number of particles, and we were extremely happy about that. So all I want to tell you today, Michele, is that in the last couple of years, we went even further. I hired a postdoc from computer science, and in 3 months, he came into my office and he said, what you did in your postdoc is nice, but I can do it better. So, and he reduced the scaling to even quadratic. So we can actually do bosonic PMD simulations of thousands of particles now. And then another brilliant physics MSc student came, and we thought, okay, let's do this also for periodic systems. That should be pretty straightforward, no? But if you want to do periodic systems, then it turns out that in every spring, each one of those lines is one of the springs of the ring polymer. You have to sum up of winding around the periodic box of the cell. And if you try to do this, even defining a cutoff, let's say you only allow it to wind up to twice around the box, even if you allow it to wind up to twice around the box, you need to do this for every permutation of the n factorial ring polymer configurations. And again, I ended up working on an even worse exponential scaling algorithm. So the only thing I want to tell you about this, Michele, is that we just published a paper where we can take this scaling, also this scaling down to linear with the winding cutoff and quadratic with the number of particles, and maintain about today. And that is about stochastic resetting for enhanced sampling. So I'll try to break it down for you. How many of you heard about stochastic resetting? Enhanced sampling, everybody here knows. Stochastic resetting? Oh, well, you unfortunately have to listen to me. You've heard me not so long ago. I'll tell you all you need to know about this. And the work has been published in these list of papers and has been carried out in collaboration with my brilliant colleague at Tel Aviv, Shlomi, and my brilliant PhD student, Ophir Blumer, who was brave enough to take up this project. And if I'm out of time, everything I'm going to tell you about today was recently published in our review in Wire's Computational Molecular Science, which is called Have You Tried Turning It Off and On Again? Where is Garrett? Do you know the reference? Everyone in the UK know the reference? From the IT crowd, no? The show? Okay. If you haven't seen it, you have to watch it. Fine. What is stochastic resetting? So I'll start with diffusion under stochastic resetting. It's a paper that came out about 15 years ago, and basically, I think, started the whole field in statistical physics. And they considered, right in 2011, a particle diffusing in one dimension from minus infinity to plus infinity. What could you possibly learn new about diffusion in one dimension in 2011? You could. So if the particle sits at the origin and needs to reach a target, which is a distance L apart, how long will it take it to reach the target on average? It's written right over there. The first moment of the first passage time distribution is going to diverge. It's going to take on average an infinite amount of time to reach the target because you can get lost to minus infinity. These two guys, Evans and Majumdar, what they did is they looked at what this distribution looks like when you periodically take the particle that is diffusing and reset it back to the origin. And they showed that once you do that, the first moment, the mean of the first passage time distribution, no longer diverges. Every time you start the process, there's going to take a different amount of time of reaching the target, so there's going to be a first passage time distribution. Stochastic resetting is taking such a process and periodically restarting it, resampling an IID copy of the initial conditions distribution. The resetting can be done at fixed time intervals or can be done at times that are themselves sampled from some distribution, okay? This is what stochastic resetting is. And why would you want to do this? So consider this case where you have a particle going from bottom to top, but there's an unreactive state that you can spend a lot of time in it without reaching the target. Stochastic resetting, just like in the diffusion example, would then limit the amount of time you can get lost and might help you reach the target faster. But it doesn't always work. Luckily, my colleague at Tel Aviv, before I even got interested in this, derived a universal sufficient condition for resetting to accelerate any stochastic process. And this is the condition. You just calculate the first and second moment of the first passage time distribution without resetting. And if the standard deviation is larger than the mean, the coefficient of variation is larger than 1, then resetting is guaranteed to lower the mean first passage time. So my friend told me about this, and I thought, hey, can we get a free lunch? Can we accelerate molecular dynamics simulations just by restarting them? And the answer is yes. This is the first take-home message. Resetting accelerates molecular dynamics and metadynamics simulations. First, another thing I learned from Michele is that you start with model systems to know what you're talking about, then you try to build up the complexity. So I'll try to do that. But it won't be as complex as Max Bonomi, right? Or, okay. You take some model systems, and they're simple enough so that we can run the brute force sampling and calculate the first 2 moments of the first passage time distribution without resetting, okay? So they're long, but not too long, so you can't do it. And you calculate the coefficient of variation, and indeed you see that it is larger than 1, okay? So that is guaranteed that resetting will lead to acceleration. And then we do the simulations, fixed time intervals or at time intervals that are sampled from an exponential distribution. And then we said, okay, can we do this not just for model systems, but for real molecules? And we looked at alanine dipeptide. And also here, it's simple enough so we can run the simulations and calculate the first passage time distribution, although already this is quite challenging. And you can see the first passage time distribution without resetting in blue. It is not really exponential. It has an exponential tail, but it has at least 2 slopes here, you see? Some trajectories finish very quickly, but a lot of them take forever to complete. And that's why we call this a rare event, okay? Once you add, how does resetting accelerate this sort of process? Once you add resetting, it is a little counterintuitive. Those trajectories that were already going in the right way, about to complete very quickly, those take longer with resetting. You see that part where the green squares are a little higher than the blue, circles? Those get delayed by resetting. But the slowly decaying tail of the distribution, resetting limits the amount of time you can spend before reaching the target. So you get, in the end, with resetting, an exponential process that on average takes less time than without resetting. Okay. So we did this, and it was nice, and I told this to my friend Shlomi, and then I told him, but we, in enhanced sampling, the entire field of resetting, they know exactly what happens without resetting. And all they want to do is to predict what would happen with resetting. But our whole community is the exactly other way around. We can't sample the process without resetting, it's too slow, so we try to use all these tricks to accelerate the sampling. And I told him, though, what do you do to recover the unbiased kinetics? And he said, I don't know, we never did it. It wasn't an issue in our simulations. So we came up with a way of inferring the unbiased kinetics from simulations with stochastic resetting. And the key point here is that resetting doesn't hurt the natural dynamics of the system up until 1 over the resetting rate. All that time scale is completely free because resetting does not bias the dynamics below 1 over the resetting rate. So all of those points you get for free. You do just 1 set of simulations. And then you extrapolate to having done no resetting at all. How well does it work? Well, it's an extrapolation. So there's a trade-off between the speed up you get, how far you start your extrapolation from, and the accuracy you get. But this is, for example, the trade-off. In blue you see the speed up, and in green you see the predicted mean first passage time. And all of these speed ups are giving the right mean first passage time to within an order of magnitude. So you can try to use this to extract kinetics if the speed up is not too high. In fact, I will tell you later we can do better, but I will get to it in a second. Then after, this was the first paper. After we did this, we said this is nice, you can do this for molecular dynamics, but only 1 order of magnitude is not enough. But the beauty of resetting is that you can do it to any kind of stochastic process. So you can also do this to metadynamic simulations. The question, is it worth it? So this is what we did. We took, again, back to model systems. You need to go from the right-hand side to the left-hand side. And in this system, if you just do resetting in blue, you get an acceleration by a factor of 5, you know? Nothing to write home about, but who wouldn't want to work 5 times less hard for their simulations, right? Now what happens when you do metadynamics for this system? For this system, you know what the collective variable is. It's just the x-axis. And when you know what the collective variable is, metadynamics always leads to faster acceleration because you know where you need to push to get to the target. So that's the green squares over there. It works better than resetting if you know what the collective variable is. But what happens if you add resetting on top of that metadynamic simulations? 2 cases. Either resetting doesn't hurt, which is shown here in the middle, the orange triangles, or resetting gives another order of magnitude on top of the acceleration by metadynamics. Why? Because, well, I'll tell you why in a second, but this is just an outcome. You can actually accelerate metadynamics as well, because resetting doesn't care what the stochastic process is, as long as the condition hit, and metadynamics stops working, okay? You see this in the green squares. Metadynamics stops working when you rotate the collective variable. What happens when you add resetting on top of it? You get the orange curve over there. So adding resetting to a poor collective variable works almost as well as using the optimal collective variable, okay? So does this hold? And the reason is that when you have a bad collective variable is you're not only pushing in this direction, you're also pushing in non-conducive directions that don't lead to the product. And resetting limits the amount of time you can push in those directions. So does this work also for a more realistic example? So we took chignolin in explicit water and we tested. We took several CVs that existed in the literature. I'm showing here 2, but we tested several others. Which 2 did we take? One is RMSD from the folded state, the simplest collective variable you can think of. And the other is Giovanni's multi-state HLDA, right? So they worked harder to, it's the simplest data-driven possible approach you can have, but it's still something you need to run trajectories in both states to get it, right? It's more sophisticated than the RMSD. What does resetting do? And you can see that HLDA is a good collective variable, shown in green in the middle panel. It leads to speed up of about 200. But on the right-hand side, RMSD is a horrible collective variable. Doesn't lead almost to any speed up, right? You see that even if I increase the bias deposition rate, it doesn't get faster. What happens when you add resetting on top of it? Well, for the good collective, either it doesn't hurt or it helps a little. Again, who wouldn't want to work twice less or 50% less for their simulations? But for the poor collective variable, it helps a lot. And you can, again, also for a more realistic example, work with pretty poor collective variable and add a little bit of resetting. It works almost as well as working with the optimal collective variable, as we all know. Resetting is most efficient in acceleration of a flat potential landscape. So the combination of flattening with a poor guess of the collective variable and adding resetting actually leads to an acceleration that is almost as good as working very hard to find the optimal collective variable. That's what we're trying to say. Okay, and I don't have time to tell you about this because I want to use my time to say in the next 5 minutes to tell you about the newest stuff we're working on, but resetting can actually also improve the kinetics and inference from infrequent metadynamics. It's an approach we call the short-time infrequent metadynamics, where you just focus on those short time scales that aren't affected by resetting, and it was published in JCTC, and thank you, Omar, also for mentioning it, and I urge you to have a look there as well. It also improves the inference of kinetics when you combine it with metadynamics, with infrequent metadynamics. Okay, the last thing I want to tell you, I think I have 5 more minutes. How am I doing? More or less? Yeah? Yeah. I want to tell you about something that came up in a conversation with Giovanni Cicotti when he was in Neve Ilan in Israel in a conference. I met him for a beer and something really nice came out of it, and then it turned into something completely different. And that is that so far, when you reset your simulation, the resetting process is completely agnostic to the actual stochastic process you're interested in. So it doesn't matter if you were just about to reach the target, if the time comes to reset, you reset, okay? This is prediction of exactly when it will accelerate and by how much. But it is also counterproductive, because if you were diffusing at the bottom well and already going towards the target, let's say you had one resetting event, now you're going towards the target, if it's time to reset, you're going to reset. It knows nothing about the reaction progress, standard stochastic resetting. So it can lead to resets even when simulations are close to the target. But what happens if we incorporate some information on the reaction progress in the resetting protocol? Let's say the simplest way to do this was define some it work. So again, we took chignolin in explicit water. This time, we didn't start from the unfolded state going to the folded state. Rather, we started from a misfolded state here in the middle, going to the folded state. And why is this an example where this sort of information helps? Because there are many misfolding, unfolding events before you actually go to the folded structure. So what you want to do is you want to reset your simulations whenever you're going from the misfolded to the unfolded state, but no resetting at all when you're going from the misfolded to the folded state. So that's what we did, we defined a cutoff of an RMSD of about 4 and only reset if the RMSD was larger than 4, okay? How does it work? You can see here in red, when you just do metadynamics with the RMSD, very poor acceleration. In green, with standard stochastic resetting, like I told you before, you get much better acceleration. And if you include just this naive way of including a little bit of information, you already get much, much higher speedups in the mean first passage time by just including a little bit of information. But then the idea is, what do you do when things are not so simple? Like, how do you know what sort of information to include? And even in this case, how did we know where to put the threshold, okay? So that, the question is how to choose the best rate and the best threshold, how to incorporate this information in a smart way. So that, and I won't tell you everything about it, I'll just tease you a little bit, and I'll tell you that the problem was that there was no theory of resetting that depends on the state and time. Reach the target until the last leg where you successfully reach the target. And because this property of resetting, that every segment is in the details, which can be obtained by just reweighting simulations without resetting. So if you have a little bit of sampling without resetting, let's say you run some metadynamics with some poor collective variable, you can reweight those simulations and obtain a prediction of what the mean first passage time will be under any state-dependent and time-dependent resetting protocol. And this is what we did. The final thing is that we then represented this resetting rate that depends on the state and time as a neural network. We defined a loss function, which is the mean first passage time to reach the target, and we optimized the state-dependent resetting rate that will lead us fastest to the target. So this is what you see here. The last slide I'm going to show. These are the two learning curves at the top right here of both simulations starting from unfolded going to folded or from misfolded going to folded. Yellow is misfolded, blue is unfolded. And these are the learning curves. What the predicted speedup is as a function of the number of epochs. So you see there are two plateaus in both cases. So what has the network learned? What sort of resetting rate has it learned? In the first plateau, in both cases, it learned a constant resetting rate that knows nothing about the reaction, just like I showed you before. But then it kept learning, and it learned the yellow and blue curves here, which are non-trivial functions of the RMSD. So it can learn what function of, in this case, the RMSD, the resetting rate should be to lead to the highest acceleration and fastest speed up. So this is the thing I'm most excited about, because we can do this not just for the mean first passage time, but also for the resulting non-equilibrium steady state that you can get. So now we can use this as a knob to design what the non-equilibrium steady state you will get under the resetting would be, and I leave you with just these nice pictures. So to conclude, it is a different way of accelerating molecular dynamics simulations. You can get between and it also works best when you combine it with metadynamics, and if you combine it with poor collective variables, it might work as well as that machine learned collective variable you've been working on for months. It might not be needed. Just try resetting, it might be similarly well. And the thing I'm most excited about is that you can actually learn now these informed or adaptive rates to optimize how your process behaves under resetting, be it the mean first passage time or the resulting non-equilibrium steady state. Thank you again, Michele, really for everything. I think I became a different scientist after interacting with you, and I can never thank you enough for it. So thank you all for listening, and I'm happy to take it.
Thank you. So, time for quick questions.
Fascinating talk. 2 questions. I might have missed it. Do you have like a recipe for finding the optimal starting or resetting time? And then do you have an intuition for the extrapolation, the form of the extrapolation?
Yeah, so if you have, the beauty of resetting is that if you have the first passage time without resetting distribution, you can predict everything you want about the process with resetting. So, and this is now also true for any state-dependent resetting. Now, if you don't have any sampling, you're in trouble. What we found is helpful is that if you have a guess of the mean first passage time, you take slightly more than that, and then it works about the rates you want to use and not have to guess. You get what the rate should be to begin with. Yeah. And a prediction of what the speed up will be. Wait, but there was another part. What was the second? So the extrapolation, we haven't optimized it yet, because now what we do is we use this
question is what happens if you happened to start in the bad well? You know, what happened if you had started the original trajectory on the right-hand side?
Yeah.
And, you know, in the one, the movie, the little movie that you have.
Yes.
Yeah.
Okay, so the answer is that we treat it in the following way. The initial condition distribution is a part of the stochastic process for resetting. So if you started at a delta function on the right well, I don't expect it would lead to much acceleration.
But it will make it worse.
It would probably, it can also lead to deceleration. It doesn't work all the time. It depends on the initial condition distribution. But if you don't know what the states are and you start from a bad state, then this won't help. Yeah, it could happen. It's not always helpful, and it depends on the initial condition distribution. But when we tested delta functions versus Boltzmann distributions, we didn't see huge differences. You could also use that.
Okay, thanks again, Barak. Our last speaker.
I forgot the microphone too.
Can you hear me now? Yeah, okay. So, good afternoon, everyone. I'm Timothée Devergne. I'm a postdoc in the team of Michele, and the work I'm going to present today is the result of a collaboration between the team of Michele and the computational statistics and machine learning team of IIT, which deals mainly with transfer these concepts to bias simulations. Then I'm going to show our results on models and applications and on to real-life intrinsically disordered protein. And finally, I'm going to show our latest work, which deals with the blind exploration of metastable states. So what does it mean studying the dynamics? So basically, we are interested in transitions between states, and we want to know at time t, what is the probability of finding the system in each state. And we do this because in experiments, what they have is not just a simulation box with a single protein, but they have many molecules in different states. And so what they obtain is not this protein is at this state, this protein is in this other state, but more the occupation number of each state. Meaning that we get the probability at time t that the system is in each state. And so we need a probabilistic view on using molecular data, using data from molecular dynamics to study time evolutions of system. So, so what does it mean statistically to study in which state is the system? It's basically studying the occupation number of the states, and the occupation number is just the integral over the states of the instantaneous probability distribution, and this one distribution. So here, if you know PT, basically you can have all the occupation numbers of your system. There is one framework to study this occupation number and this PT, which is the Koopman or the transfer operator, depending on your community, that can be obtained from unbiased simulations. what it does is that instead of studying PT, you study the deviation of the instantaneous probability distribution with respect to the invariant Boltzmann distribution. And then what the Koopman operator does is that it propagates ut from time t to time t plus tau. And so if you have an initial probability distribution, what you do is you apply n times the Koopman operator, and you know this ut at time n tau. And so you can see the evolution of your system and the occupation numbers from ut using this Koopman operator. So there have been a lot of work on this operator and a whole zoology of studies. But all these studies, they require the evaluation of the time lag correlation function, which is this C of tau, which requires the evaluation of an observable or of a function at time t and at time t plus tau. And as we will see, it can cause some problems. Because, for example, here it's still a figure from an experimental paper, and you see that their experiment lasts 140 minutes. And so, as it has been mentioned a lot of times today and yesterday, these reactions cannot be sampled in one single molecular dynamic simulation. So we need some form of enhanced sampling scheme to do this. But this will reduce the dynamics at which we have access. We will not be able to have access to UT. Because what you get from a single bias simulation is basically the dynamics. And if you perturb the dynamics, the correlation function that you get, C bias of tau, is different from the C bias of tau. And the question that we can ask is, okay, we have something that depends on tau, and what happens if I make tau go to 0? Is there a differential equation that describes the evolution of UT? And actually there is, if we place ourselves in the Langevin dynamics assumption. Basically, we will assume that the probability distribution relaxes towards equilibrium according to a Langevin dynamics. And if we do this, there is a partial differential equation, which is the backward Kolmogorov equation that was shown by Christophe 2 talks ago. And this equation is without here the periodic boundary conditions. It's a bit cumbersome because it involves second derivatives. You need to know the potential. But the nice thing is that it linearly depends on U. So to overcome the fact that we need to solve a PDE, we can rely on a model decomposition of the operator. So what we are going to do is to decompose the operator into eigenfunctions and eigenvalues. And the eigenfunctions and eigenvalues of this operator have nice properties because they are all positive and they're growing. And if you know the eigendecomposition of this operator, you can recover, for example, UT. And each of the eigenfunction and the eigenvalue, they correspond to transition between the two main wells. And then the two others, they translate the transitions in between the two subwells. And then by doing this eigenfunction decomposition, we have transformed a PDE into an eigenvalue problem. Now the question is, how do I learn these eigenfunctions and this eigenvalue decomposition? So the problem is, given a biased trajectory, how can we learn the eigenpairs of the system? So the first trick that we use is, instead of learning directly this infinitesimal generator, we will learn the operator that is called the resolvent, which is basically just shifting and inverting this operator. Because as we have seen, the slowest the mode, the closest to 0 the eigenvalue will be. So you will try, if you learn directly L, you will try to learn things that are very close to 0. Instead, if you learn this resolvent, you will have the interesting eigenvalues, which will be away from 0, and all the rest that will be close to 0 and that you don't care about. And as we have seen in the equation, L involves second derivatives, but it has some nice properties because there is this Dirichlet form that allows transforming the inner product between two functions and simply an integral over the product of the gradients of the functions. So here, with this trick of the Dirichlet form, you don't have the potential appearing, you don't have second derivatives, so it's a much simpler problem to use, and you only have a Boltzmann average to compute. So now, how do we learn usually an operator? Given a set of function phi I, you will try to find the matrix G in the span of the phi I that will approximate the best the operator Boltzmann averages to compute. You don't have anymore this correlation function with tau in this. So this means that if you have Boltzmann average to compute, you know how to rewrite it from a bias simulation, and you can compute the average easily. Now the problem is, okay, we have this basis function, but can we learn them instead of just using fixed basis function? And actually, we can learn them, and we will parameterize them with neural networks. And with a simple loss that involves only Boltzmann averages, and the lambda here is just a matrix, a diagonal matrix containing the eigenvalues that you want to learn. So they are also parameterized and learnable. And the main message here is that here the time dependence of the Koopman operator is replaced with the differential operator, and you don't have any time dependence, and you only have Boltzmann averages to compute. So this means that we have a pipeline for learning our operator. So given an initial bias data, you need to characterize your system with descriptors. Then you learn this phi I, which are called a representation. You regress the generator with the closed form, and finally you can compute the eigenpairs of the system. So what happens with application? So we started with the traditional example of alanine dipeptide. We had two data sets, one which is based on phi, which is a very good collective variable, and which leads to many transitions, and psi, which is a very bad collective variable and gives not a lot of transitions. And what happens if we learn the generator on these two data sets? We have information about the transitions dynamics. So until now, I talked a lot about dynamics, about time evolution of occupation numbers. I did not show you any result on this. So how do we do to obtain this kind of occupation numbers? So our method is that you start your system in one metastable state. So basically what happens when it's in that metastable state, it is restricted to the Boltzmann distribution in that metastable state. And if you compute U in this state, it's just the ratio of partition numbers. So you have your initial probability density, and you can propagate it using the generator. And then if you have UT, you can basically obtain also occupation number by integrating. And also what is nice about the eigenfunction is that they can give us an idea about the states because it changes sign around the transition state. So in one state, the eigenfunction will be positive. In the other state, the eigenfunction will be negative. And for alanine dipeptide, we started it in the C7AX state, and we let it relax towards the two other states. So, okay. But it's a very simple system with only basically two main states. So can we do it for systems with several states? So we went to a slightly bigger molecule, which is alanine tetrapeptide, which has several modes, and also, its slowest modes are characterized by the two angles phi2 and phi3. And when we do this, we learn the eigenfunctions, and here we see that it allows us to identify which states are which mode to propagate the dynamics and get information about the time evolution of the occupation numbers. So we started this in the top right state, which is the least stable one. And we propagate it using the generator. So here it's not a simulation, it's just doing Boltzmann averages and applying the generator. And what we observe is that starting from this state, it will start filling the other state that is the nearest one. And then this state, which is not very stable either, will also populate the two other. And this allows us to actually identify that this other state is an intermediate. So until here, I showed you only the result for only small molecules. But what happens for bigger molecules? So we have this collaboration with the company Pepton around intrinsically disordered proteins. There was the talk of Massimiliano Bonomi on intrinsically disordered proteins, so I will go very quickly on this. But they are very interesting for health reasons. But what happens with intrinsically disordered proteins is that they don't have two metastable states, two very distinct metastable states. So here I put the picture of a simulation of one IDP, and we cannot see anything from it. We cannot identify any clear states. And can the generator help us identify the states? So our guinea pig was amyloid beta 42 behavior of the system, and so they give us a low-dimensional space because we have maybe the maximum we can have is 10 slow modes. So they give us really a low-dimensional space in which we can analyze the data, and so we used Gaussian mixture to do clustering, and we found basically 4 states. Okay, so here I plot the free energy surface and the points of the simulation with the clustering. But what guarantees me that the clusters I find really correspond to structural changes? So let's look at the secondary structure. And actually, what we see when we look at the secondary structure, here it's the proportion of helicity. The first eigenfunction corresponds to actually the breaking of a helix around the residue 25, and the second eigenfunctions also correspond to the breaking of another helix. And this is actually nice because we have a way of getting information from a very blurry, a very complicated simulation, and we can obtain really the clear states of this simulation. And so the eigenfunctions, they allow to associate each mode with the structural changes, and they really, what they do is that they provide a low-dimensional space in which we can work. So the main ideas of this method is that it is only based on Boltzmann averages. So if you have a method that is not molecular dynamics to generate Boltzmann distributed sample, you can use it. So if you have Monte Carlo, you can use it. If you have, for example, a generative model, you can use it. So it's applicable for any kind of data generation scheme, as long as your samples are Boltzmann generated. We have I hope it will work. I don't know what happens because it's still there, but it's not on the projector. Oh, okay. No. Because either it's on the projector or there, so... the HDMI cable, yeah.
Stable connector, I think. Which format is your slides?
PDF.
Can we transfer from our
Yeah,
computer?
yeah. Okay.
Don't touch.
Yes, it's good.
Because maybe the signal is too low. Yes. Yes, could you go completely high? Yes.
No, it's not changing
there,
anything there. Yeah, sorry about this.
It's very quick. Oh, your video board has some issues.
What happens?
No, it's disconnected. It's just your...
Yeah, so...
Something happened.
Maybe no... Maybe if I unplug it... Oh, let's... No. Again for this computer.
Do you have any drive, online drive with the files or not?
Yes, but it will take some time, so... No, but I will finish yeah. Maybe if you plug it on the other side.
Maybe. It doesn't like maybe this port.
Yeah, no. Well, so, yeah, so really sorry about this. So we had a last, a new paper that we submitted one month ago, basically, which is on discovering metastable states from only an unbiased simulation. And what happens is that we train this generator model on only this the one state, and what happens is that the first eigenfunction, which is the trivial one, which should be constant, it's constant on the training set, but then it deviates from its constant value. So our idea was, okay, can we use it for exploring the chemical space? So what we did is that we used this curvature using the gradient of the system. Yes. So if you have a double well potential and you only have data here, the eigenfunction will basically look like this. It's constant here, and then it goes like this. Because it's the extrapolation region of the system. So what we did is we used a bias that is minus the logarithm of the gradient square of the constant eigenfunction, plus. And what happens is that it actually lowers the barrier. So the effective potential will be something like this. No, actually. Something like this. So it will directly go to the other state with only a simulation there. And we applied it to, so double well potential is not the most interesting system, so we applied it to alanine dipeptide, to chemical reactions, and to alanine tetrapeptide. And so I guess I can finish here.
Thank you, Timothy. And also thank you for drawing your presentation. So, questions?
Very nice talk. So I was wondering, so for the A beta 42 system, how do you determine the number of states or number of clusters that you should get?
So by optimizing it, you can see the number of clusters that you have.
Thank you.
There further questions? No? If that's not the case, let's thank Timothée again and all the speakers of this afternoon session.